Good morning, everyone. Welcome. Welcome to the 12th annual ICCI conference. And there's two microphones here, not because I'm in stereo, but because we're recording this, live streaming it to YouTube as well as recording it for later. Not all the talks, but certain ones that people have agreed to. A few little housekeeping things. There's another group in the cafeteria. So we're supposed to keep to the right side. So the wrong side would be the dairy group. Nothing wrong with them, but there's a whole dairy group in there. And we're on the poster side. So the whole cafeteria is taken up with conference today. So you're welcome to take your coffee and snacks and lunch even back in here if you need extra room. But you can also stand in there and admire the great posters, which would be good too. I'll just tell you that this is all possible through the Arthur Willis Visiting Professorship in Canine Oncology and support from the OVC Dean's Office to run this thing with all the food and the posters and everything else. Anything else we have to say? I think that's it. We have the very, very last speaker couldn't make it today, so we have a little bit of extra buffer on time at the very end before our keynote, which is more time for coffee break or questions, of course, and discussion. Okay, so I'll let Michelle introduce our first speaker. So you'll have to put both mics on when you come up. It's a bit of a pain.
Yeah. Okay. Wonderful. So our first speaker this morning is Dr. Jim Petrik. Jim is a professor in the Department of Biomedical Sciences and also adjunct faculty at McMaster and Western. And he leads a collaborative research program to discover novel therapies to treat advanced stage ovarian cancer. His long-standing collaboration with Dr. Jack Lawler from Harvard has resulted in a development of a mechanism to normalize tumor vasculature and enhance an uptake and efficiency of conventional therapies. Jim is extensively published and has been very successful in achieving amazing research grants, and he also sits on review panels for both CIHR and the Israeli Cancer Research Fund, and we're very excited to welcome him as our first guest this morning.
Awesome.
Yeah, seriously.
Awesome. Nice. Awesome. I feel like a cyborg now. This is good. I'd just like to start by thanking Jeff and Michelle for the invitation to come present some of our work and the rest of the ICCI committee. So we're really excited about sharing some of the work that we've been putting together for the past 15 years or so. So as most people know here, our lab's looking at ovarian cancer and some ways to address advanced stage ovarian cancer, which has some really particular challenges to it. So just by way of introduction, epithelial ovarian cancer is the most lethal gynecologic cancer. It has a very low 5-year survival rate, and its median age is around 65, so it's a postmenopausal condition. So there's some hormonal implications that we're looking at as well, but we'll talk a little bit about some of the vascular issues as a focus for our research. So EOC is a stage disease. So stage 1, disease is localized primarily to the ovaries. In stage 2, disease has migrated to the pelvic basin, but it's still contained within that area. In stage 3, it's metastasized now and spread transcolomically throughout the abdomen. And by stage 4, there's distant metastases. The numbers under here are the predicted 5-year survival rate if the woman was diagnosed at that stage. So if a woman's diagnosed at stage 1, it's almost a curable disease. The unfortunate thing is most women are not diagnosed until stage 3. Because of the very vague, nondescript clinical symptoms that go along with ovarian cancer. So at this stage, there's typically a very advanced miliary disease, metastatic disease throughout the abdomen. So there'll be a surgical resection of the primary tumor and then typically high-dose chemotherapy with platinum and taxanes. The challenge for ovarian cancer is that there's been no real therapeutic advancements and no real change in outcomes in 40 years. So we've taken on the challenge of looking at advanced stage ovarian cancer. That's the current clinical picture. So we're interested in looking at other mechanisms, other therapeutic interventions that we can develop that can help improve these outcomes. One of the things that our lab's really interested in looking at is tumor angiogenesis, which is the formation of new vessels from pre-existing vasculature any tumor that grows beyond 2 millimeters cubed is required to recruit its own vascular supply. And this is done by changing the balance between promoters of angiogenesis and inhibitors of angiogenesis. And I don't have enough time to go into a lot of the mechanisms, but some of the important promoters, vascular endothelial growth factor, which are referred to as VEGF, and thrombospondin is the inhibitor. This is the mechanism, the tool that we've been focusing on for the past decade or so, is optimizing ways to use TSP1 mediated approaches to help reduce this angiogenic drive. So as I mentioned before, a small tumor can exist happily and receive everything it needs through passive diffusion and osmosis. But in order for it to grow to become a more clinically relevant situation, it needs to recruit its own vascular supply. And it does that by very potently secreting pro-angiogenic factors. We talked about VEGF as one. There's a whole host of other ones. And so it floods the area with these pro-angiogenic factors. Adjacent vasculature will respond to this and start to sprout and mobilize vascular tubes towards chemotactically towards the tumor that was secreting the pro-angiogenic factors. But the problem is the tumor doesn't really know how to regulate the process. It says, I need a vascular supply, so it really aggressively floods the system with pro-angiogenic factors. The vascular supply responds to that in a very aggressive manner because of the level of pro-angiogenic stimuli that's in place. So the result is that although there's a tremendous number of blood vessels that form in the tumor, they're actually very poorly organized, they're very poorly formed. So we end up with a very dysfunctional, disarticulated vascular supply that ultimately ends up, ironically, resulting in a very poor perfusion of the tumor. So there's tons of blood vessels there. They're not mature, so they don't actually carry much vascular supply to the tumor. So we end up with a situation. So if we look at a tumor, we end up with a very disorganized, dysregulated vascular supply. So we end up with a lot of necrosis, a lot of hypoxic regions within the tumor. And this has a problem from a number of different reasons. First off, we reduce supply to the tumor because of the vascular supply being so dysfunctional. We get very little vascular supply to the tumor. Because the tumor is growing and we have cellular proliferation and increase in size, we get increased pressure within the tumor. So increased interstitial fluid pressure collapses lymphatic vessels that reside within the tumor. So now we're not getting fluid in, and we can't drain the tumor because the lymphatic vessels are being collapsed and compromised. So we have a real challenge in getting things into the tumor. From a therapeutic perspective, if you think most chemotherapy, most cancer therapies are delivered intravenously. So we're relying on an incredibly dysfunctional system to get our therapies into the tumor. And I'd liken this tumor to my teenage boy's brain. So, you know, their brains are already completely filled with knowledge and wisdom and everything, so it's jam-packed, so we can't get any more information into the tumor or into their brains. Same thing happens with the tumor. Okay, there's this tremendous amount of pressure, really reduced delivery opportunities, so it's really a significant challenge to get anything into the tumor. So as Michelle mentioned in the intro, I've been working for a long time with Dr. Lawler from Harvard, and he was the one that cloned thrombospondin 1 back in the day. Thrombospondin 1 is an amazing peptide, large protein. I can't really get into its biology, but it does a lot of really intriguing things. But the problem is it's a really massive protein. It's a 450 kilodalton glycoprotein. So it's not really a good therapeutic tool. So what we've done is we've isolated the type 1 repeat region out of the thrombospondin protein, I'll call it 3-TSR, and it does all of the awesome things that we want TSP1 to do from an antiangiogenic perspective. It binds to its receptor, CD36, it has glycosaminoglycan binding domains, activates TGF-beta, so essentially it inhibits a lot of the pro-angiogenic stimuli. So we initially hypothesized that 3-TSR will specifically target dysfunctional tumor vasculature and this result in this vascular normalization to reduce the presence of those dysfunctional, non-functional vessels in the tumor. So initially, in order to study this advanced stage ovarian cancer, we had to develop an animal model to replicate it. So it's a simple model where we orthotopically inject 1 million spontaneously transformed surface epithelial cells under the ovarian bursa. Mice are really convenient in that they have a bursal sac that lines the ovary. So we just inject 1 million cells under here. Those cells then can interact with the ovarian microenvironment. And we published a couple of papers showing that interaction with the microenvironment is actually critical in normal disease progression. These cells are p53 wild type. When we inject them, the cells that metastasize in the abdomen have developed a p53 mutation, which actually really closely replicates what happens in women. So the women, the cells derived from the ascites fluid in metastatic disease are p53 mutant, and we've been able to replicate that. So our disease model looks like this. We end up with animals that have large abdomen due to ascites or fluid accumulation within the abdominal space. They have large primary tumors, and then they develop metastatic disease through transcolonic metastases. The tumors are high-grade serous adenocarcinoma, which replicates about 90% of the epithelial ovarian cancer. So we've developed a model that has been a really good workhorse for us. It replicates the disease. We've staged it. We can allow animals to get the different stages and initiate treatment. So we've had a lot of work done with this model. So initially, we just want to see, does 3-TSR have any efficacy in tumors? So we initiated tumor formation. We started treating with 3-TSR, and we found that if we look at this tunnel assay, the green dots are the dying tumor cells. When we co-localized with CD31, which is indicative of endothelial cells, we saw that there was specific targeting of endothelial cells as well. So we saw that we could increase apoptosis both in the tumor cells, and we could reduce blood vessel density following 3-TSR treatment. So there seemed to be a fairly potent anti-tumor and anti-angiogenic effect. And I'll come back to that because we think that that's part of the impact of this therapy is it has a bimodal effect. It's cytotoxic to tumor cells, but it also has functions within the vasculature as well. So if we look, this immunofluorescence staining is looking at CD31, which is staining blood vessels in red. The green stain is for alpha smooth muscle actin, which is staining the smooth muscle support cells that line mature blood vessels. So blood vessels, the last stage of angiogenesis is actually maturation, where the blood vessel, the tubes have formed, but very importantly then, we need to have smooth muscle cells that line and support those structures. And what that does is it provides infrastructural support, also provides cell communication with endothelial cells, which conveys a survival signal, but it ends up really regulating morphology and function of the blood vessels. So if you look in the top, the control treated mice that just received PBS, they have extremely large, really awkwardly organized blood vessels, okay? Whereas in 3-TSR, we've gotten rid of those really large dysfunctional tumor vessels, and we end up with very small, luminal, kind of appropriately formed or appropriately shaped vessels. So we've normalized the vascular morphology. And then when we look at tumor hypoxia, we've reduced the level of hypoxia in the tumor. So by inference, then we've enhanced perfusion to the tumors by normalizing and remodeling the vasculature. Which I'm not going to lie, when I say the hypothesis, where we said that we were going to preferentially target abnormal tumor vasculature, that was not our original hypothesis. I was trying to make myself look smart, that we actually predicted this was going to happen. This is a totally unanticipated outcome. We thought we were going to destroy blood vessels and starve the tumor in a traditional anti-angiogenic approach. At the end of the day, what we did is we shrunk the tumors down, we made them small, but we gave them an amazing blood supply. And initially we thought, this sucks, right? Like now we're giving the tumor everything it wants. But what we found out is that, if we enhance delivery to the tumor, we already talked about before that delivery to the tumor is one of the major impediments to therapeutic efficacy. So if we can't get stuff into the tumor, how is it going to have its effect? So then we thought, well, is there any way that we can exploit this improved perfusion to enhance drug delivery? And we did a simple experiment where we just radio-labeled a chemotherapy drug. We injected it, and we saw that after vascular remodeling, we saw a significant increase in the radioactivity of the tumors, so a lot more of the drug got into the tumor. So this was our first kind of inkling that we may have an opportunity to enhance delivery to these tumors. Which is awesome, but completely clinically irrelevant. We need to look at advanced stage disease. Like I said, that's the clinical picture. So starting treatment at the initiation of tumor development isn't relevant. So we developed a model where we allowed mice to get sick and replicate about stage 3 disease. So they had large primary tumors, they had metastatic disease, they had the initial formation of ascites, and that's when we started treatment, because that's really the clinical situation. So what we did was we allowed them to go to about day 80, they had stage 3 disease, and then we started treatment with 3-TSR. And this was a really exciting result for us a little while ago now, but we saw that in the control group, this is what the tumors looked like when we started treatment. In the control group, they grew from this to this. But in the 3-TSR treated group, not only did they not grow to this size, but they actually shrunk. So this was an indication that with a 3-TSR, just as a monotherapy, it was able to induce regression of advanced stage disease. So we were pretty excited about those data. So then we wanted to look at, if we can optimize delivery to the tumor, can we now use this in combination? Can we use 3-TSR to normalize the vasculature to optimize or increase the efficacy or uptake of chemotherapy drugs? Most women and most individuals undergoing cancer therapy, regardless of what solid tumor type they have, undergo what's known as maximum tolerated dose, which means they get an extremely high level of delivery of the chemotherapy drug. They're given a drug-free interval to recover and recuperate, and then they're given subsequent dosing. For women, most women undergo a 6-round initial MTD of carboplatin paclitaxel. Extremely devastating on the individual from side effect perspective. There's more recent work being done on metronomic chemotherapy, and there's a number of comparative oncologists here that are looking at metronomic chemotherapy in companion animal models, where we give smaller dosages, smaller dosages, but there's a chronic administration frequency. So we never allow the individuals to exist in this drug-free environment. The problem with this drug-free environment is this is where disease resistance occurs, or therapy resistance occurs. So in the initial administration of high dose, we may have cytotoxic death of a lot of the tumor cells, but the residual ones, that's a time where they can develop their resistance mechanisms. So drug efflux pumps and a whole host of other mechanisms to avoid the next onslaughts of chemotherapy administration. Metronomic chemotherapy reduces the problem of having the drug-free intervals. But the problem with metronomic chemotherapy is if you give really small doses, how are you going to get enough drug into the tumor? That's the reason why they have to give maximum tolerated doses. You give a whole crap load of chemotherapy drug in the hopes that some's going to get it into the tumor and have the cytotoxic effects that you're looking for. So we thought, look, if we've enhanced or increased efficiency of delivery, maybe we can optimize and allow enough chemotherapy drug to get in using the metronomic approach. So what we did is a simple experiment is we utilized 3-TSR on its own, maximum tolerated dose chemo, and metronomic chemotherapy alone or in combination with 3-TSR. And complicated slide, but what I'd like to just draw your attention to is that 3-TSR, when we've allowed the molecule to induce that vascular normalization, combine it with low-dose metronomic chemotherapy, it had the most potent anti-tumor effect, which it also had a very potent antivascular effect, reduced the microvessel density. But then when we did survival studies, when we looked at the controls, all the control mice were dead by 100 days. Chemotherapy drugs on their own improved survival, but then anytime we added 3-TSR, we saw that we had a fairly significant increase in survival. And then the group with the 3-TSR and metronomic chemotherapy, out to 150 days, which is half of a mouse's lifespan in some cases, we'd only lost 1 mouse due to disease. So we were really encouraged that this combination of vascular normalization and this low-dose metronomic chemotherapy had some really interesting outcomes. So the main findings for that is that when we did the vascular normalization, we really increased the efficacy of these outcomes. So then we asked, can 3-TSR be combined with other treatment approaches? And this simulated the collaboration with Sarah Wooten and Byron Bridle from Pathobiology, where we looked at oncolytic viral therapy. So oncolytic viruses, and I'm not going to go into this too in depth, first, mostly because I'm not capable, but oncolytic viruses have a preferential cytotoxic effect on tumor cells. Normal cells typically are impervious to oncolytic viruses, where tumor cells are susceptible. But the problem with oncolytic viruses is that almost universally they cause a vascular shutdown. So you can get some oncolytic virus into the tumor, but then they cause a vascular collapse, which is a real problem because if you want any subsequent therapy uptake, if you want to allow immune cells to get into the tumor after oncolytic viral stimulation, that's a real challenge because of this vascular shutdown. So we want to see if 3-TSR is really good at normalizing the vasculature and maintaining patent delivery to the tumor, can we overcome that vascular shutdown that's induced by the oncolytic virus? So what we did is an experiment. The oncolytic virus that we were using is Newcastle disease virus. So 3-TSR on its own, once again, shrunk the tumors. And this was done in the advanced stage again. So we allowed the mice to get to about stage 3. So we induced regression. Newcastle disease virus on its own also induced regression, but then the combination very potently regressed advanced stage disease. This arrow here, what I'd like to draw your attention to is if you look at this tumor, it's really blanched. It's an incredibly pale tumor, which really highlights and supports this theory that the oncolytic virus on its own causes this vascular shutdown and reduces vascular supply to the tumor. So, and if you look here in the combination, we can see that it apparently, in a gross perspective, is a reperfused tumor. So we've been able to combat that vascular shutdown, reestablish perfusion. At the same time, we're still inducing regression. Okay, so it still has a cytotoxic effect while allowing vascular supply to the tumor. So this is, in blue is hypoxia, in red is endothelial cells, and in green are the smooth muscle cells. So if we look, there's a lot of hypoxia in the PBS treated tumors. 3-TSR induces this vascular normalization. NDV, you can see rampant hypoxia. Very limited vascular supply to the tumor and a complete absence of mature blood vessels. So this is really kind of indicative histologically of that vascular shutdown that we hypothesized was going to happen with the oncolytic virus. And then when we combine, we see nice small luminal blood vessels, significant reduction in the level of hypoxia. So and once again, these tumors are really small. So we shrunk them down, we gave them good blood supply, and we dramatically increased the amount of oncolytic virus that was able to get into the tumor. But one of the main functions of oncolytic viruses, when you look from an anti-tumor perspective, is they have a direct cytotoxic effect. But one of the main effects that they have is that they stimulate intratumoral immunity. So they increase the recruitment and activation of NK cells, of cytotoxic T cells. So one of their main jobs is to go in and start inducing lethal cytotoxicity, but also to stimulate that immune system so that you can have intratumoral immunity. But the problem is, if you recruit the immune cells, you need to be able to get them into the tumor. And there's some really cool pictures out there that following oncolytic viral therapy and that vascular shutdown, you can see immune cells lining the periphery and the perimeter of the tumor because they can't get in. So they've been activated and recruited, but they can't get access to the inside of the tumor. So this is a really incredibly complex slide, but what I want to highlight is that PBS, 3-TSR, oncolytic virus on its own, and the combination, when we look at intratumoral presence of these immune cells, so macrophages, natural killer cells, cytotoxic, CD4, CD8 positive T cells, we can see that in combination, when we normalize the vasculature, let more oncolytic virus get in, and then maintain that highway for the immune cells to get into the tumor, we can really increase the number of immune cells that get into the tumor. Okay, and we have some other data to go along with this showing the efficacy and the survival. These mice survive for a long, long time after we've done the combination therapy. But now what I want to talk about for the last couple minutes is where we're going next. So 3-TSR is awesome, but the problem with it is it's a small peptide and it's cleared from circulation in about 8 hours, in about 14 hours, sorry. So that's all right. It definitely has efficacy and it has some biological function, but the problem is women are not going to want to go in for daily intraperitoneal administration of a therapy. So we've looked at developing a novel compound where we combine 2 3-TSR peptides with a non-immunostimulatory human immunoglobulin. And what that does is it increases the half-life from 14 hours to 8 days. So now this could be a weekly administration of a therapy, which is far more clinically relevant. And the other thing was we've seen FC3-TSR, which is the nomenclature for this fusion protein. So FC3-TSR also has increased biological activity. And it's through this concept of receptor clustering, where if you put 2 of the 3-TSR molecules, and we've seen this for other FC peptides, you get a clustering of the receptors. So you get increased biological responsiveness in addition to the prolonged half-life. So we're increasing time in circulation of a drug that does better things that we've seen with native 3-TSR. And we just did some initial experiments looking at the tumor outcomes. So if we look, we did different dosages, and the mid dose of FC3-TSR significantly reduces tumor weight and metastases, importantly, compared to 3-TSR on its own. So we saw 3-TSR did really fantastic things, but it looks like the fusion protein is even more potent. So it has more in vivo potency. We've shown a whole bunch of different in vitro efficacy that's better in FC3-TSR than 3-TSR on its own. And we looked, it's better at remodeling the vasculature, so we see an increased proportion of these mature pericyte-covered or smooth muscle-covered vessels in the FC3-TSR treated group. And then when we look at drug uptake, once again, we just radio-labeled chemotherapy drug, we see that FC3-TSR fairly significantly increases uptake of the drugs, even in comparison to 3-TSR, which we showed had significant in vivo efficacy. So lastly, one of the things that I talked about initially was that the increased interstitial fluid pressure collapses those blood vessels, or sorry, lymphatic vessels, so we can't drain the tumor. So that's important from a fluid drainage perspective to reduce the pressure, but also the intratumoral immunity, if we increase the uptake of or activation of checkpoint inhibitors, anti-CTLA4, anti-PD-1, PD-L1, all of those things need to get to draining lymph nodes in order to further educate the immune system and enhance the anti-tumor immune response. And I'm not showing the data today, but we've been able to kind of reanimate the lymphatic function in these tumors. So if we look we're just looking at a stain that stains lymphatic endothelium in the tumors. In PBS treated groups, which has really reduced vascular supply, okay, you can see reduced vascular supply in the tumors. We have lots of hypoxia, and we have reduced presence of intratumoral lymphatic vessels. Whereas in FC3-TSR treated in the green, you can see there's lots of vessels that are still contained within the tumor core, which facilitates drainage, but also once we activate the intratumoral immunity, then we can now allow for those checkpoint inhibitors to drain to the lymph nodes where they really need to get to. So the working hypothesis right now, just in conclusion, is that an untreated tumor has really big, large, nasty, dysfunctional vessels, lots of hypoxia. We have collapsed lymphatic vessels, which really reduces their ability to populate the draining lymph nodes with immune regulatory molecules. Okay? When we treat with FC3-TSR, we reduce the presence of these dysfunctional vessels. We end up with small, luminal, smooth muscle cell covered functional vessels. We reanimate the presence of lymphatic vessels. We allow the uptake and activation of a whole host of different immune cells, NK cells, cytotoxic T lymphocytes, and then we facilitate draining of the relevant molecules to the drain lymph node, once again, kind of really kickstarting and amplifying the immune response. So that's where we're at right now. We're taking this, we're hoping to take this to human clinical trials within the next year or so. We've developed a small startup venture to attract venture capital to mobilize towards that. But the other thing that we're interested in doing is bringing this to companion animal clinical trials here at Guelph. I've talked about ovarian cancer, and ovarian cancer is the model that we've been looking at in the lab. But this is not restricted to ovarian cancer. All solid tumors that require vascular supply, which is basically all of them, should respond in a very similar fashion, especially other vascular hormonally responsive tumors. We're thinking of some of the soft tissue sarcomas, hemangiosarcomas, things like that, should respond very similarly to this. So with some of our awesome companion animal oncologists here, we're trying to establish some collaborations, doing some initial safety trials, and then move to companion animal clinical trials with this as well, because the real strength of this is that any therapy that you need to get into the tumor, so chemotherapies, oncolytic virotherapy, immunotherapies, putting in FC3-TSR initially to normalize the vasculature should increase uptake and efficacy of those tumors. So we're excited to try those out in some of our hard to treat companion animal tumors as well. So the real take-home message of this is that drug delivery with this abnormal dysfunctional tumor vasculature is met with a whole host of different delivery problems. We can't get the therapy where it needs to go. Okay, we increase the efficacy and efficiency of delivery. That really should help increase the potency of current conventional therapies. And I'd just like to end by thanking everyone in the lab that's done a lot of work over the past more than decade on this. In particular, Kathy's been working on a lot of the FC3-TSR work, so she's done some really good work on that. I'd like to thank the collaborators here at Guelph, looking at the oncolytic virus and immunotherapy perspective. Great thanks to Jack, who's been a fantastic collaborator on this for the last long time, and then obviously funding. We really appreciate that. And thanks for your attention. I'd be happy to take any questions. Yes.
Jim, thanks for an excellent talk. Stupid plant biologist question, so I'll preface it with that. Just to ask, do you get any off-tumor effects? That is, 3-TSR has an effect on normal angiogenesis, so what happens in the rest of the body that's honestly, that's the important million-dollar question. And so what we found is that even in the most aggressive physiological angiogenesis, so wound healing, the angiogenic mechanisms built in that whole maturation as the blood vessels are developing. We found that FC3-TSR and 3-TSR on its own is the blood vessels that have smooth muscle cell coverage are impervious to it. So we've done wound healing assays where in our animal models, we've induced wounds and we've given them high dose FC3-TSR, and it does not impede normal angiogenic mechanisms. So the wounds heal at the identical rate. We've done retinal corneal vascularization assays. We see no impediment or impairment in normal angiogenic functions, which is important because a lot of our normal physiological processes, reproductive processes, especially in females, ovarian cycle, menstrual cycles are very dependent on normal vascular function. So we're really encouraged that we don't seem to be derailing that. Yes?
As far as the combination with metronomic chemotherapy, the doses, metronomic chemotherapy, the primary target is in the cancer cell anyway, so there must be something else going on that's causing the synergy that you're seeing.
Yeah, absolutely. And what we think is that, and we haven't looked at this, we're going to go back and do these experiments because we weren't thinking of it at the time as the immune response. So metronomic chemotherapy, I agree, is cytotoxic to the cancer cells, but Bob Kerbel's done a ton of work on the immune responsiveness to metronomic chemotherapy. It has some antiangiogenic effects. We think that we're kind of trumping that a little bit with the FC3-TSR, but I think it's the immune response to metronomic chemotherapy that we're helping to accentuate. Brenda.
Tim, I'm going to on the record and say we all know that the pipeline is clogged with failed antiangiogenic therapies.
Yep.
And the interesting thing about ovarian cancer, as you pointed out, is that the cancer cells themselves, the neoplastic cells themselves, rely on VEGF signaling. So I'm wondering if this dual effect is the real key reason why 3-STR works. And have you thought about looking at it in other models where there is neoplastic expression signaling, like melanoma, for instance? Before you start saying it's going to be universal antiangiogenic,
right?
Which probably won't actually.
So you're absolutely right. So the thing is, if you look at it from a just solely an anti-angiogenic perspective, it's kind of boring. I agree. Like you look at Avastin, Avastin has had not awesome effects, which targets the VEGF pathway. But I think the secret to why we see efficacy here is that it has completely non-VEGF related functions. So if we take the VEGF side, we put it head to head with Avastin before, and it does way better job because of its specific effects that signal through the CD36 receptor. We found that any tumor cell, and we've done a bit of a panel, any tumor cell that expresses CD36, which is receptors, they're quite responsive to it. So we think that that's the key, is that tumors that express CD36, and we're going to do a panel of the companion animal tumors to identify expression. Most of the epithelial tumors are CD36 positive. We're just starting to branch out now. But I think that's the key, is if we divorce ourselves from just the specific VEGF-mediated effects, it still has a lot of efficacy mediated just specific through that signaling pathway through CD36. Jeff?
The ovarian model and the actual disease is a bit unique in that it's transcelomic metastasis.
Correct.
And some of these antiangiogenic therapies have failed because they increased metastasis, but that's all hematogenious, right?
Yep.
I'm worried that that's gonna, you found something that works, but it's working in a very unique situation.
Yep.
Do you think any evidence that it doesn't increase metastasis by?
No, it's a great question, right? The one thing we've done, it's still an ovarian cancer model, is remove the primary tumor, and it still has efficacy in the metastatic disease. Where sometimes if you remove the primary tumor, metastatic disease just goes rampant. So still is able to put the brakes on that and induce regression in that system. But no, we haven't. We're just starting to branch out. There's been a lot of work just in this model. We're now starting to branch out to, we think it's, prostate cancer is going to be relevant. Basically, it's very similar to what we see in ovarian cancer. But no, distant metastatic disease, we can't replicate metastatic, like hematological metastatic disease, stage 4 disease in the mice because they die first, as most women do. So it's a great question, and we need to start working in other models. We just need some grant funding to do that, which is the bane of all of us.
Just to follow up on that, I was wondering, same sort of thing, most of your data is on the primary tumor. Do you see the same vascular effects in the metastatic, or are they too small?
Yeah, no, it's a great question. So we see identical things in metastatic. So all the if you can see the tumor, it's big enough that it needs its vascular supply. So we get some fairly large metastatic tumors, and they've completely developed their own vascular system, and we see the exact same thing. And Kathy's done a bunch of work where we've taken out the metastatic tumors, and we see increased uptake, increased delivery of immune cells, increased chemotherapy drug uptake in the metastatic tumors as well. So I think that's why it's particularly effective in metastatic disease, which is really the critical thing. The primary tumor, although we use it as a model, it's really a surgeon's problem. No offense. But the metastatic disease is what ends up killing the patient. So we're more interested in does it have an effect on the metastatic disease? We use the primary tumor just to show biology. Alicia.
It's a mechanistic question. So your peptide is inhibiting, is destroying the vasculature that is abnormal, or is it also increasing the recruitment of pericytes?
No, it does. We don't think that we haven't really looked at, it's a good question because we haven't really looked at PDGF receptor and other things that stimulate parasite. What we do see is it induces a really rampant apoptotic cell death of those non-parasite covered. So we think it's completely a pruning of the abnormal vasculature rather than a maturing of existing unmature vessels. I don't think that it does that. I think it just specifically goes in and wipes out the abnormal vessels.
So do you know if the domain that is activating TGF-beta, which is included in that peptide, have any function in the antiangiogenic?
I don't know. To be honest, I'm resistant to get into TGF-beta because I'm not smart enough to, it's way too complicated. I hate it. Jack has an interest in TGF-beta, and I don't. So we don't know what the TGF-beta mediated effects are doing. We've done some TGF-beta mutants where we've knocked out the TGF-beta activating domain in the 3-TSR, and it really reduces the efficacy. So we think that, we don't know why that is, but it seems like there's some TGF-beta mediated effects, but we haven't had a chance to pursue it. Right. Yeah, absolutely. And we actually thought that by mutating it, we'd increased efficacy, but we did the opposite because TGF-beta never behaves as you think it should, right? So we don't know. We haven't pursued it.
I have one more question.
Yes.
I understand that it's really early on in the stages of the clinical application. But any preliminary data on safety?
That's what we're just hoping to be able to do that initially in a healthy cohort of companion animal patients, just to do the safety data. I can tell you in our preclinical model, we've seen no indication of any cytotoxicity or off-target effects. So the animals that we give even very high doses to are still reproductively competent, which means that their vascular function is still maintained. We've done the wound healing assays. We see no other sequelae in our preclinical models. We want to do it in companion animal healthy cohort, which then with those safety data in hand, then we can move to companion animal clinical trials and the human clinical trials as well. Awesome. Thank you.
Hi everyone, so my name's Elaine. I currently work in Dr. Byron Bridwell's lab in the Department of Pathobiology here at the University of Guelph. So today I'm going to talk a little bit about our combination therapy using Decitabine, which is an epigenetic modifier, and oncolytic virotherapy. So I'm kind of going to touch on Thomas's virus a little bit, but he's already done too good of a job explaining it. So before I start, I just want to give an acknowledgement. My best friend Jaden actually passed away in March of 2017. He actually passed away after a 7-month battle against acute myeloid leukemia and 3 relapses. So that's kind of why I got into blood cancer research and why I ambushed Byram for the grad studies. And so I just wanted to leave an acknowledgement slide. So with that being said, I'm quite familiar with the current standard of care in human medicine. And typically, so just to show a little bit about kind of how strenuous this can be on patients, this little boy, Oliver, he's featured here with his beads of courage, sometimes referred to as beads of bravery. So each bead depicts a different event in their treatment regimen. So for example, one girl went into isolation and she got a bead with a teddy bear. Another boy he started to lose his hair, so he got a bead with someone basically balding. Another girl traveled on a train for some of her chemo treatments, so she got a train on that bead. So it just shows really how many treatments and events during their treatment they have to undergo right now. And with that being said, typically the current standard of care starts with a minor surgery. So they'll get what's called a central venous catheter, sometimes referred to as a Hickman. Goes in at the chest area, and basically it prevents patients from having to constantly get new needles to get blood drawn and to get IVs in. It is a minor surgery, but still a surgery nonetheless. Next, they'll undergo remission induction. Because they're looking to treat acute leukemias, they are quite aggressive. Typically, they go into remission induction almost right away. With Jaden, it was about 24 hours from his diagnosis. And they call it 7 and 3. So basically, you'll get 2 types of chemo drugs. The first one being something like, for example, Cytarabine for 7 days. And the last 3 of those 7 days, they typically like to give anthracyclines, such as doxorubicin or daunorubicin, for example. And after you're in remission, it typically takes it can be up to 2 months in hospital, depending on the patient. And then you'll get consolidation therapy. So this can be a wide range of things. It can consist of radiation therapy. It can consist of mutation-targeted drugs, like if you have the FLT3 mutation. It can also consist of low doses of chemotherapy, which is one of the more common ones. And this is just to keep patients in remission. Next, many leukemia patients will have to undergo a stem cell transplant. This can either be allogenic or autologous. Allogenic transplants typically have a bit higher of a survival rate at the 5-year mark. The problem is that if you don't find a match that's related to you, you need to be a match on 12 human leukocyte antigen markers, I believe something like that. So it can be hard to find a donor if you don't have siblings that are a match to you. So sometimes they can also do autogenous transplants, and they're doing really cool things actually with ex vivo therapies. But basically what they'll do is they'll actually take the stem cells from the patient, they'll purge them ex vivo of leukemic cells, and they'll reintroduce it back to the patient. So it's important to appreciate the drugs that we use. So before, oops. Okay, that was supposed to be a time-lapse video that was going to play, but it doesn't appear to be working. But basically, we use two types of therapy or reagents. Sorry, one of which Thomas has already covered, oncolytic viruses. So I'm not going to go over them. This was supposed to be a time-lapse video that basically showed cells expressing GFP as they get infected. And I only showed the first 24 hours, but if you watch the full 72, you can actually see the cells die off, which is pretty cool. It also allows you to visualize it in real time. And we also use epigenetic modifiers. So as Thomas alluded to, this is one thing that's approved actually in human medicine for monotherapy for acute myeloid leukemia, albeit with limited efficacy and high rates of relapse. So what it actually does is it reactivates some of the silenced genes that the cancer actually mutates and basically hypermethylates. So it inhibits the methylation of newly synthesized DNA. With that being said, it only works on proliferating cells, so it does not target the cancer stem cell niche. But some oncolytic virotherapy does. It can target cancer stem cell markers. So it's also important to appreciate the type of cancers that we're treating. So I'm going to be referring to them as T-ALL and AML throughout the presentation. T-ALL being T-cell acute lymphocytic leukemia, coming from basically the T lymphocytes shown here in the lymphoid lineage, whereas AML comes from the myeloid lineage. So getting into our treatment regimen, we start by giving, depending what we're trying to actually induce in the mice, if we want to give them T-ALL, we'll typically give about 100,000 EL4 cells. If we want to induce AML, we'll typically give about 1 million C1498 cells. Again, those were just optimized to be a lethal dose in controls. Typically, we'll wait 7 days for the leukemic cells to engraft so that the mice are actually tumor-bearing, or rather leukemia-bearing. And we'll give Decitabine on day 8. So Decitabine goes intraorbital. It does have a very, very short half-life. So we do give it in 2 doses. We wait 24 hours and we give it again. Now, in order to separate the treatments a little bit in time, we do give NDV 3 days later, although that's not a huge separation. That's just kind of what we had optimized treatment for. And we basically push the dose at about 1.0 times 10 to the 8 plaque-forming units of NDV. So that's how all the following data was generated. And as you can see, our combination therapy induces durable remissions in both models of leukemia. So you can see here up top in the AML model, we get survival out to about a day 110 here. These mice actually were rechallenged at day 60. I'm not going to show the data, but I'm showing some VSV rechallenges next. But as you can see, it does achieve quite durable remissions in both models. And next, anybody who works in leukemia and the type of area there, you'll know that relapse disease is huge. As I mentioned, Jaden relapsed 3 times in 7 months. So we really wanted to know if our treatment is indeed conferring resistance to relapse disease in rodent models. So what we actually did was we rechallenged mice that survived the initial treatment. And again, this is using a different oncolytic virus. This is using VSV, courtesy of Rob Mold. And basically, you can see 100% of the mice survived. Of course, we'd want to do this with a few more mice in a bigger scale study, but it is quite promising results thus far. So next, we wanted to see kind of how strong the memory response was. So we would normally inject 100,000 EL4 cells into mice. Instead, we re-challenged them with 1 million cells. So we put quite a leukemic burden on them, and we actually still had a 40% survival. But what we wanted to do then was see if this immunological memory was in fact leukemia specific. So we actually took these survivors here, and we re-challenged them with 1/3 of the dose of AML cells. So we took our T-ALL survivors and we re-challenged them with AML, so a different type of leukemia. As you can see, they didn't really survive much longer than controls. So it is a leukemia-specific immunological memory. In literature right now, it's already been reported that Decitabine pretreatment sensitizes cells to oncolytic virotherapy. So what we wanted to do was, instead of simply pre-treating cells and then doing the type of Resizurin assay that's usually done, we actually took mice that were treated with Decitabine monotherapy that were at endpoint. So the drug should have been completely out of their system at this point. And we basically collected we harvested the brains of these mice, and we collected these brain-derived Decitabine surviving cells, and we separated them out using the RAF flow cytometer in pathobiology. And we figured out that indeed the phenomenon does hold true. They are still more resistant to, or more sensitive to, oncolytic virotherapy. So something we actually want to do in the future is to go in vivo with the brain-derived EL4 cells and see if it actually changes our survival statistics. So we reported a different mechanism of action. So prior to this, literature reported that the Decitabine surviving phenomenon in the Decitabine pretreatment was due to defects in type 1 interferon signaling, which is very common. The virus can get in much easier with defects in interferon signaling. We actually report that it's a increase in reactive oxygen species production. So Decitabine actually upregulates reactive oxygen species. So we generated Decitabine surviving cells that basically killed off 90% of them. Only the 10% that were left were the Decitabine surviving cells. We cultured those up for a few weeks, froze them down, and used them for these experiments. So what we wanted to do was we actually wanted to test this. There's a few lines missing. We actually wanted to test this in vivo. So what we did was we actually co-administered a pan-reactive oxygen species scavenger known as N-acetyl-L-cysteine. And the two important lines here, I guess you can see, basically almost completely abrogated therapeutic efficacy. So when you do scavenge up all those reactive oxygen species, it does have a significant impact on your therapy. And something I want to do in the future is actually dissect which reactive oxygen species are responsible. So we've ordered the reagents to do different assays for this and basically fine-tune versus a pan-reactive one. So in conclusion, now it's important to take this with a grain of salt and realize we've only done this in mouse models. So the current standard of care slide that I presented at the beginning was human medicine. This slide is more depicting what we've achieved in mouse trials. So we basically have 2 doses of Decitabine, 1 dose of oncolytic virus. It's 3 days total of injections, 4 if you include the engraftment cells. But basically, Decitabine is clinically approved as monotherapy in human trials, which is why we're so interested in that, because it's a lot easier to come in with 1 novel therapeutic than 2. And we actually achieved durable remissions and resistance to relapse disease in mouse models, as seen. And then just in conclusion, I want to give a little bit of acknowledgement to my advisor, Dr. Byron Bridle, seen here on our picnic day, and my lab team, in particular, Jacob Van Vloten, Dr. Megan Strachan Whaley, and Dr. Khalil Karimi, who personally mentored me throughout the course of my grad degree, as well as our virus production team, Thomas McAusland, who just spoke, and Dr. Lisa Santry, and our animal care staff, who this would absolutely not have been possible without. And I'll take questions.
Questions for Elaine?
So where do you see this going from a comparative approach? There's certainly been national trials in the U.S.
Yeah, so I mean, one would hope it pushes through and gets approval, but our next steps, we're kind of looking to work with Paul and the Animal Cancer Center, hopefully push this through to some companion animal trials, and as Jim mentioned, kind of get safety in those trials. We've already worked out safety details in mice, but it'll be a different story when we go into dogs if we get there, so that's kind of the goal by the end of my grad degree.
In your long remission mice, did you ever see evidence that there was in fact brain leukemia cells that are sort of dormant in those mice?
We actually, so after treatment with, are you talking about the Decitabine monotherapy or the combo?
Any of them.
So in our combination, we actually did flow cytometry at endpoint, or we euthanized them at a certain point in time. And I didn't show the data here just for time restraints, but we actually showed that there were no residual leukemia cells. So we use a congenic marker to find them, but we actually showed in our combination there's no residual leukemia cells. In the monotherapies, there are residual leukemia cells.
So if you left those monotherapy mice long, do you think those cells could recur and induce a student disease?
Yes. And I mean, again, the problem here is, right, Decitabine's in trials right now, but the problem is that it has such high rates of relapse out past like the 5-year mark, right? And the problem with oncolytic viruses is they work great in solid tumors, but with leukemia, it's spread throughout normal tissues that can quench viral infections, right? So I do think if those monotherapy mice were left, I do think they would get relapse disease, which we've actually shown in our survival curves. They don't survive. Every single one of them dies when our combinations survive.
But that might be a really good model to see if oncolytic viruses by themselves can deal with that relapse disease.
We've tested that they can't. Yeah, so we've done tons of survival studies, and again, we're really pushing that dose of oncolytic virus just to the point of where it's not cytotoxic. So we give fluids every roughly 3 hours to 6 hours. So we couldn't also push the dose any higher, which is something we wanted to try, is even lowering it. And eventually at some point we want to try some kind of viral mimic like poly-IC to see if maybe it just needs to think there's virus there. It doesn't actually need a virus itself.
Do you viruses cross the blood-brain barrier?
I believe the student prior to me did that research, but that was before my time, so I'm not sure.
Maybe equally or...
Yeah. I know stuff was done with that, but it was the PhD student prior to me, so.
Given that cytosine methylation is important in terms of memory formation, my understanding is that some of the aversion to using epigenetic modifiers like Decitabine in human treatments is because of cognitive impairment. Do you see any behavioral differences in your mice that have had the Decitabine treatment?
In combination, no. In monotherapy, yes. There's a lot of cognitive issues. They start to get hind limb paralysis, stuff like that. Their body kind of doesn't really function properly. But in combination, we actually don't see any cognitive deficits, which was interesting. Right.
All right, so I'm going to jump track a little bit and talk to you about bacteria. So thank you. I'm Avery. I work in the Alan Virka laboratory that primarily focuses on the human gut microbiome, and my research focuses on Fusobacterium nucleatum and its role in colorectal cancer. And today I'm going to be presenting some very preliminary data on the potential to target virulent colorectal cancer-linked Fusobacterium nucleatum with a predatory bacterium, Dellavibrio, and like organisms, or BALOs. So colorectal cancer is among the most commonly diagnosed and lethal cancers in Canada, and its incidence is expected to rise in adults under 50, as well as globally by 2030, due to the adoption of the Western lifestyle spreading throughout the world. Additionally, this is a particularly lethal disease, and one of the reasons for this slide changing isn't working. It's just delayed. So one of the reasons for this is that it's particularly difficult to diagnose this disease at early stages. This is significant as late-stage diagnosis of colorectal cancer is linked to poorer overall disease survivability. So this is the stage of diagnosis, stage of colorectal cancer at diagnosis in Canadian men and women between 2011 and 2015. And you'll note that almost half of them are diagnosed at later stages. And later stage is linked to a relatively poor 5-year relative survival rate. So colorectal cancer is further complicated by its complex etiology. There are a number of risk factors associated with developing this disease, including ones that you can control, so your diet and your alcohol intake, and things that you can't, like your age and your genetics. And further, this disease is complicated by the proximal colon microbiome. And the human gastrointestinal microbiome is known to provide a lot of key benefits to us, like expanding our metabolic repertoire and outcompeting opportunistic pathogens for physical and metabolic niches, but it has been noted to be skewed in colorectal cancer patients, and it's very complicated in terms of the metabolic processes that might contribute to disease progression. Fusobacterium nucleatum is one such member of the human gastrointestinal microbiome. Specifically, it resides within the oral cavity. It's an anaerobe, meaning it doesn't grow in the presence of oxygen, and it's gram-negative, meaning it has 2 cell membranes instead of just 1. It's a very heterogeneous species in terms of its genotype and its phenotype, and it's also known to be a commensal. So normally it doesn't do anything particularly beneficial or particularly harmful in our mouths, which is great because we likely all have this species. But it can be virulent, and it's considered to be an opportunistic pathogen. It's considered to be an opportunistic pathogen specifically in colorectal cancer, among other diseases and infections. So this link was first found in 2012. 2 separate North American research groups, one of which included the Alan Virka lab, found that Fusobacterium nucleatum was more abundant in colorectal carcinomas compared to adjacent healthy tissue within the same patient. Since this discovery, there have been a number of different discoveries in terms of Fusobacterium and its link to colorectal cancer, including it's considered to be a disease passenger, meaning the disease is already present when Fusobacterium nucleatum arrives and promotes the pro-inflammatory and pro-cancerous state. Its presence has also been linked to poor disease survivability. So patients in a European colorectal cancer cohort were noted to have a poor disease survivability with high levels of Fusobacterium in their lesions compared to patients with low levels of Fusobacterium nucleatum in their lesions. Additionally, another research group in 2017 found that Fusobacterium in primary colon tumors as well as metastatic liver tumors, suggesting that this bacterium not only survives for extended periods of time within the colon tumor environment, but could also travel with the tumor as it metastasizes. So I'm now going to jump track a little bit more and talk about Dellavibrio and like organisms, or BALOs, and their microbiology. So they're a really interesting bacterium because they're gram-negative and they're small, even by bacterial standards, fitting through a 0.45 micrometer filter. They're also very highly motile, and these prior to characteristics contribute to their predatory capabilities. They're very successful at predating other gram-negative bacteria. In particular, they're useful for biofilm clearance, potentially with the ability to clear biofilms more effectively than antibiotics. And additionally, they are bacteria specific. They don't elicit any extensive damage to eukaryotic cells, and they've also been used to treat animal infections thus far, I believe in cow eye infections. So this is the unique life cycle of Dellavibrio and like organisms, or Balos. Their life cycle starts with being in an attack phase where they're motile, they're looking for a prey, and then they find one. They attach, they penetrate, and they establish themselves. And when they establish themselves, that is within the periplasm between those two membranes of their gram-negative prey cell. Then they grow in what is called a deloplast, causing the prey cell to round up. And following that, they septate, so they divide into individual cells, they develop, they grow their flagella, and then they lyse their host cell and go out on the prowl for further prey. So the first question in this research is, well, Dellavibrio is well characterized in terms of its predation in model organism E. coli, but can it prey upon gram-negative Fusobacterium nucleatum? So there have been only 2 studies thus far suggesting that it can, and we wanted to know specifically, can it prey upon our inflammatory bowel disease and colorectal cancer isolated strains of Fusobacterium nucleatum? So to do this, first we would prepare Fusobacterium nucleatum and Dellavibrio separately, and then co-incubate them aerobically, grow the surviving Fusobacterium nucleatum anaerobically, as it is an anaerobe, and then count the Fusobacterium nucleatum colonies. So the results are in terms of how many Fuso survived the predation and survived the aerobic exposure. So what you're looking at here then is colony counts of Fusobacterium nucleatum, either unexposed to air or the predatory bacteria, the purple bars. You're also looking at Fusobacterium exposed to air only, 3-hour control, orange bars, and Fusobacterium exposed to air and the predatory bacterium, the red bars. Now, what you should note first is that there were some strains of Fusobacterium nucleatum that did not survive the aerobic exposure very well. So that would be like 3127 and CC53, who just did not survive, as they are an anaerobic species. However, excitingly, there were a few strains of virulent Fusobacterium nucleatum that were preyed upon by this strain of Dellavibrio. In particular, we were excited about 7.1 and its extensive predation compared to the surviving cells exposed to air alone. So the previous data was from 2 biological replicates, and that's not going to convince anyone. So we decided to do some electron microscopy imaging of this predation to see if it's a real thing. So this is the strain 7.1 predated by that previous strain Dellavibrio HD100. So here you can see the remnant of a Fusobacterium cell and these tiny Dellavibrio cells with their flagella and these weird black tumor things that we couldn't really decipher by TEM, so we moved on to scanning electron microscopy. So a reminder, this is what Fusobacterium looks like. It is a long spindle-shaped cell, very thin, very tapered, and when it's attacked by Dellavibrio, it forms these, again, this cell, a remnant here, and these Dellavibrio swarming it, and these weird bolus things that we don't really know what exactly is going on here. So zooming in on this area, to us at the Biomedical lab, it really looks like Dellavibrio is stealing Fusobacterium nucleatum's outer membrane, right? It exists between the 2 membranes of Fusobacterium nucleatum to complete its predatory life cycle. You can see it is draped somewhat in a membrane here compared to this cell over here. So that the typical life cycle, again, is that in E. coli cells, the entire cell balls up to form this deloplast. So we're hypothesizing that because Fuso is so long and thin, that deloplast isn't able to form. So instead, like when you steal the sheets from someone you're sharing a bed with, Fusobacterium nucleatum's outer membrane is stolen by Dellavibrio so that it can grow within it. So the question 2 then is that Fusobacterium nucleatum is an anaerobe, and Dellavibrio is an aerobe. So how can this predation work? So to avoid stressing Delavibrio too much, we decided to give it its optimal host, its preferred E. coli, anaerobically to see if this predation can really occur. So previous studies have been done to see whether supplementation, like giving an alternative terminal electron acceptor, right? Like we use oxygen, Delavibrio uses oxygen. Can we give it something else so that it can prey upon an anaerobic bacteria? It does have several nitrite reductase genes, meaning it could possibly use nitrite. And various other studies have been done looking at supplementing Delavibrio HD 100 with glucose, with nitrate, and found that it works, but then another study found that it didn't. So the literature on this is really confusing. So thought, okay, let's just repeat it and see if we can get it to work. So the experimental protocol looks exactly like the previous one, except E. coli is the prey, and we're using anaerobic predation conditions, and we are supplementing with various terminal electron acceptors and also electron donors to potentially bolster the Delavibrio growth. So first, we're looking at nitrate and glucose. So what you're looking at here is anaerobic predation of E. coli, either with no supplement or supplemented with nitrate, supplemented with glucose, or nitrate and glucose. And what you can see is that really there was no assistance given by the addition of the supplements. There is some slight predation from 3-hour control just exposed to the filtrate of the Delavibrio without any actual Delavibrio. Compared to exposure to the Delavibrio. So potentially this means that there was some initial predation in that 3-hour window, and then the Delavibrio died. So we thought, okay, let's look at possible electron donors and acceptors that we could give to Delavibrio so it can survive these anaerobic conditions. So within the genome, there is the possibility of HD 100, there is the possibility for it to use nitrite and various electron donors as opposed to oxygen for its terminal electron acceptor. So preliminary, just this is the control. Heapes is the broth that we use, or the buffer that we use to just perform regular predations in without supplement. You do see some slight predation after 3 hours, but none following 24 hours, meaning Delavibrio likely died. Same with Heapes and nitrite, the terminal electron acceptor alone, initial predation, but then likely just Delavibrio death. But when we supplement with lactate, saw some further predation following the 3-hour into the 24-hour mark, which is entirely new. That's never been seen before with Delavibrio. It's not been shown to predate after 3 hours of anaerobic growth. And with succinate, that was even more pronounced. However, with other supplements, Formate in particular provided a huge jump in 3-hour to 24-hour predation rates, which is very, very, very new, very exciting. And in the 3 hours with glutamate, that jump is more pronounced, although the jump at 24 hours is less pronounced. So future directions for this research, because it is super preliminary, there are only 2 biological replicates, so it's really hard to say concretely what's going on. We'd like to repeat HD 100 Delavibrio predation of Fusobacterium nucleatum and confirm each of those strains that I previously mentioned with electron microscopy, as only 1 strain so far has been done. Additionally, we'd like to further confirm the electron donor and terminal electron acceptor results for anaerobic predation of E. coli, and then potentially move that into Fusobacterium nucleatum predation. And we'd also like to look at the potential of targeted predation of Fusobacterium nucleatum. So can we get Delavibrio to target virulent strains compared to commensal strains, and can we get it to target Fusobacterium nucleatum in the presence of surrounding decoy microbes, as you might see in a colonic environment? So I'd like to thank our collaborators, Dr. Robert Mitchell and Wansik Munn, as well as the Alan Virko Lab and Dr. Alan Virko, who could not be here today.
Questions for Avery?
Yeah, nice job.
Thank you.
Since your Fusobacterium colonizes the counter tumor and goes with it to the metastatic disease, thinking outside the box, rather than try and kill it, are there some ways that we can actually use Fusobacterium?
That's a really interesting suggestion. So up until recently, there hasn't really been a way to genetically manipulate virulent strains of Fusobacterium nucleatum. There's been some work done in like ATCC, well-studied, well-characterized strains of Fusobacterium nucleatum that have been deposited, genome sequenced. But as for virulent strains, like the ones associated with inflammatory bowel disease, colorectal cancer, those have been resistant to all of our genetic modification attempts. A collaborator of ours has recently determined a method to genetically manipulate Fusobacterium nucleatum, but that hasn't really, that's very, very preliminary. So potentially, but that's in the works.
Why pursue Balos over bacteria?
So Delivibrio is an interesting organism because it's so good at clearing biofilms. It actually, in some studies, have shown that it prefers biofilms, the sessile bacteria, because once it enters that predatory phase, grows within the deliplast, and then lyses, all its prey are right there. So it's really efficient at clearing biofilms, definitely with more efficiency than antibiotics, but also potentially more efficiently than bacteriophage.
Any other questions? So that brings us to our first coffee break. If we can ask everyone maybe to come back for around 10 to 11. There are posters as well as some coffee and snacks in 1701.
So our next session, we're going to have 3 short talks, and then have another longer regional keynote talk. So our first speaker is Megan Brasher from the Department of Molecular and Cellular Biology. She's going to talk about snare.
Hi everyone, my name is Megan and I'm a PhD candidate in Dr. Coppolino's lab, and today I'm going to be discussing some of my PhD research that is entitled The Analysis of Snare Regulation During Tumor Cell Invasion. So the extracellular matrix is a network of protein and carbohydrates that provides biochemical and structural support to the cell. Remodeling of the ECM is required for cellular migration and cellular invasion and is an integral part of many physiological processes like the maintenance of tissue architecture and embryogenesis. However, the progression of pathological disorders is mediated by abnormal cellular invasion, and an example of this is cancer. So we all know that the metastatic cascade is the process by which primary tumor cells migrate to distant tissues and establish secondary tumors. However, so first, primary tumor cells must be able to degrade their surrounding extracellular matrix, and one method that tumor cells use is making these F-actin protrusions known as invatopodia. So like I said, invatopodia are F-actin protrusions that reach into the extracellular matrix to help degrade it. So in order for tumor cells to maintain their invasive phenotype, they must be constantly assembling and disassembling invatopodia at the leading edge of the cell, and they do this in 4 steps. So the first step is initiation, in which we get focal adhesion-based signaling with the ECM by the recruitment of integrins and receptor tyrosine kinases. The second is assembly, in which we get actin polymerization occurring through the recruitment of proteins like cortactin and NWASP. The third step is maturation, and this is when we get the proteolytic ability achieved through the recruitment of matrix metalloproteases like MT1 MMP. So these proteases are able to degrade the surrounding extracellular matrix. And the final step is disassembly, in which these proteins are removed and recycled. So in order for these key proteins to get to the cell surface, to get to the spot of invatopodia formation, they have to first be trafficked there. So this is partly done by snare-mediated trafficking. So snares are soluble and ethanolamine sensitive factor attachment protein receptors, and they function in a variety of trafficking pathways and membrane fusion events. So we can see here on our target membrane we have 2 T snares, and on our vesicle we have a V snare. So these 2 T snares on the target membrane interact to form a trans-snare complex. This then allows the V snare to come in, forming a cis-snare complex. This brings the vesicle close enough to the membrane so that it reduces the energy required for fusion. We can then get delivery of our cargo to the specific membrane. So it's been found in our lab that by inhibiting specific snares, we actually decrease Invatopodia formation. So previously we've looked at snares like Syntaxin 4, SNAP23, and VAMP7, and perturbing these all decrease Invatopodia formation. However, the regulation of snares may play a crucial role to this process, but it's not well studied. So another snare that interacts with Syntaxin 4 and SNAP23 is VAMP2, which hasn't been well studied in our model. So we use MDA MB231 cells, which are an adeno breast cancer cell line. The reason we use them is they form Invatopodia in vitro when they're plated on an ECM-like substance. So I wanted to see if VAMP2 expression was also required for Invatopodia formation, since we know that Syntaxin 4 and SNAP23 are. So I did this by knocking down VAMP2 using shRNA, and we plated these cells onto fluorescent gelatin. So it's labeled red, and when the cells degrade the gelatin, they leave behind this like black puncta. We then can stain the cells for an invadopodial marker like F-actin, overlay the images, and quantify for Invatopodia formation. So we can see here, our parental cells are able to form Invatopodia, same with our empty vector control. However, when we knock down VAMP2 via shRNA, we get a decrease in Invatopodia formation. So this is after a 4-hour time point. So we can also do this for a longer time point. We call it gelatin degradation. So basically they make bigger spots of degradation. We quantify that. We see the same kind of phenotype where we get a decreased of these cells' ability to degrade gelatin, indicating the expression of VAMP2 is required for Invatopodia formation. We then wanted to somehow inhibit VAMP2 function, so we did this by expressing VAMP2 cyto, which is basically all of the protein, but it's lacking its plasma membrane domain. So because of this, it's basically cytosolic, it's like free-floating. It can bind to other snares, but it's not going to be found in that vesicle. So it's not going to allow for that vesicle to come in and fuse. It's basically acting as a competitive inhibitor. So we did this Invatopodia formation assay again at 4 hours. So we look at our parental cells, they're able to form Invatopodia, same with our empty vector control. However, when we're expressing VAMP2 cyto, we get a decrease in Invatopodia formation. We also use the full-length protein as our control, and these cells are also able to make Invatopodia. So this is quantified here. We can also do this for, again, a longer time point to look at gelatin degradation. We see the same phenotype, that we get a decrease of these cells' ability to degrade the gelatin, indicating that not only is the expression important, but the function of VAMP2 is important for Invatopodia formation. So it's been previously found in our lab, like I stated before, that Syntaxin 4 is required for Invatopodia formation, and this was done by a previous PhD student in our lab, Carla. She basically found that the snare complex required for this was Syntaxin 4, SNAP23, and VAMP7, and this snare complex was found to deliver MT1-MMP to the cell surface. And she looked at the inhibition of Syntaxin 4 in the same manner I did, using Syntaxin 4 cyto. She also knocked down Syntaxin 4 via siRNA and saw a decrease in Invatopodia formation, indicating the snare plays an important role. However, the regulation of snares is not well characterized, like I said previously. So this brings us to another graduate student's work, Dave. He looked for a regulator of snares, and he found a candidate called MUNC18C. So MUNC18C is a SEC1 MUNC18 protein, and they have established roles in GLUT4 trafficking. However, the roles for these proteins in a cell invasion context are not well characterized. So like I said, MUNC18C is a known regulator of Syntaxin 4. And it can bind to Syntaxin 4 when it's in a snare complex, but the method I'm going to be focusing on today is MUNC18C binding to the N-terminus of Syntaxin 4. So it's been previously characterized that MUNC18C binds residues 1 through 29 of Syntaxin 4. So we're going to use that as a competitive inhibitor in our cells. So I expressed this peptide, or this amino acids 1 through 29 in our cell lines, added a GFP tag so we could see it. And we confirmed that it was indeed acting as a competitive inhibitor. So basically, this 1 through 29 can bind to MUNC18C, which is going to decrease endogenous Syntaxin 4 binding to MUNC. So first we wanted to see if perturbing this MUNC18C interaction with Syntaxin 4 affected snare complex formation. So I immunoprecipitated VAMP2 in the Syntaxin 4 N-terminal peptide cells, and we get a decrease in the amount of VAMP2 co-IPing to Syntaxin 4. We also did this with SNAP23. And we can see here that the same phenotype exists. We get a decrease in SNAP23 binding Syntaxin-4, indicating that MUNC18C interaction with Syntaxin-4 is required for snare complex formation. We then wanted to see if this N-terminal peptide could decrease Invadopodia formation. I don't show the data here, but by expressing this N-terminal peptide, we get a decrease in Invadopodia formation as well as cell invasion. So since we're seeing these phenotypes, we're wondering if that's affecting the trafficking of key invadopodial proteins to the cell surface. So we did a biotinylation assay, and we looked at 3 different proteins that are trafficked to Invadopodia. So EGFR, beta-1 integrin, and MT1-MMP. So you can see here, in cells that are expressing our Syntaxin-4 N-terminal peptide, we're getting a decrease of EGFR and MT1-MMP to the cell surface, indicating that MUNC18C function is required for this trafficking event. We then wanted to see, since we saw that MT1-MMP and EGFR are decreased at the cell surface, we were wondering if this was going to affect cell invasion. So we subjected these cells to a transwell matrigel Boyden assay. You can see here that our Syntaxin-4 N-terminal cells did indeed have a decreased ability to invade through that matrigel. So not only does the function of MUNC is required for trafficking of EGFR and MT1 to the cell surface, but it also is required for cellular invasion. So some next steps that we're currently working on, we were able to send some stable cell lines expressing Syntaxin-4 full-length, Syntaxin-4 cyto, which I talked about previously. It lacks that plasma membrane domain, so acts as a competitive inhibitor, as well as Syntaxin-4 N-terminal to BC cancer. They injected these cell lines into immunocompromised mice in the mammary fat pads, and then they looked at lung metastasis. So we can see here in this graph, in our control, Syntaxin-4 full length, we do get a fair bit of metastasis. They're basically weighing the lungs. However, when we, in the mice that were injected with Syntaxin-4 cyto or Syntaxin-4 N-terminal, we get a decrease in lung weight, aka these cells can't metastasize as easily. They also looked at a Kaplan-Meier survival curve, and our Syntaxin-4 full lengths did not survive as long as our Syntaxin-4 cyto and Syntaxin-4 N-terminal cell lines, indicating that we're decreasing the amount of metastasis occurring with these 2 treatments. So my research is going towards looking at a smaller peptide than 1 through 29. I'm going to be looking at 1 through 15 and also 15 through 29. So a really important residue that's been characterized previously is leucine 8 in that 1 through 29, and it's been shown that it's required for binding to MUNC18C. So by looking at the residues 1 through 15 and 15 through 29, we're wondering if a smaller peptide can bind to MUNC18C. So we did a couple experiments. First, we looked at Invadopodia formation like before. You can see here that when we express 1 through 15, we get a similar result as seen previously with the Syntaxin-4 N-terminal, that we get decreased Invadopodia formation. However, we don't see this in the 15 through 29. We also subjected these cells to a Boyden transwell matrigel assay as well. We see a similar phenotype as the Syntaxin-4 N-terminal, and we get a decrease in cellular invasion. So to kind of summarize, Invadopodia formation requires trafficking of key invadopodial proteins to the cell surface, and kind of a working model that we're thinking is occurring. We have our 2 T snares at the plasma membrane. We have Syntaxin-4 and SNAP23. And then, so upon MUNC18C binding to Syntaxin-4, we get trans-snare complex formation. This allows for VAMP2 to come in and bring in the vesicle, which allows for the transport of our EGFR and MT1-MMP to Invadopodia. So I did show that inhibition of VAMP2, SNAP23, and Syntaxin-4 decreases Invadopodia formation, gelatin degradation, and cell invasion, and that the Invadopodia formation is mediated by the snare Syntaxin-4, VAMP2, and SNAP23, but regulated by MUNC18C. So I'd just like to say a couple thank yous. Thank you for ICCI for allowing me to present. This is my 2nd time, and it's been awesome. My advisor, Dr. Mark Coppolino, my past and present lab members, if their name's in bold, they contributed to this work, my committee members, and well as our collaborators at BC Cancer. Thank you.
Questions for Megan?
Can I just go back to the graph where we show the mice that you injected in the mammary fat? So you are showing Decitabine is important for myeloid formation.
Yeah.
And you are seeing there that you get like a delay in metastasis or reduction in metastasis, but nevertheless the mice are dying at the end, right? So what do you think is happening? Do you think something can take over for Signasim before one is gone to avoid for myeloid formation, or that the cells are adapting and going without myeloid in the process?
Either could be possible. So there's a multitude of snares. So like I said, there's VAMP7. I'm talking about VAMP2, but VAMP7 has also been showed to be required for invasive formation. So there could be a somewhat redundancy there, where just because it doesn't have, the formation of syntaxin 4 complexes, it could be other ones that eventually come in, or they could be using a different method of metastasizing. Yeah, so something to look into. Thank you.
You mentioned earlier that the MUM proteins are involved in group 4 trafficking.
Yep.
Is there any relevance to that? And do you think maybe glucose metabolism is involved in the formation of snares?
That I don't know. I have nothing I really looked into. When you look into literature, everything that seems to happen in GLUT4 trafficking doesn't really seem to apply here. So it's hard to make those connections. So yeah, something to look into. Thanks.
The next speaker is Alicia Delory. She teaches faculty in biomed. Many of you will know her. Her student is off to Boston, so couldn't be here to present. So Alicia is going to present.
Okay, so I guess they go in different sides, so are they okay here? Why do we need to? Oops. Nope. Like this. Okay, so thank you very much for the organizing, to the organizing committee for inviting me here to talk today about the work that my students, Mackenzie Wong and Anita Liu, both of who abandoned me today. So I will be talking about the characterization of extracellular vesicles from culture explants of canine osteosarcoma and normal bone. So why are we interested in studying this in canine osteosarcoma precisely? Well, this is a real clinical problem in veterinary medicine. It's the most common primary bone tumor in dogs, to begin with. It affects mostly large breeds. And most commonly affect the long bones. The standard treatment includes limb amputation as well as chemotherapy, and that could be usually carboplatin, doxorubicin, either of them. And the overall survival rate, despite of this type of treatment, which is aggressive, is only about 50% of the patients surviving 1 year. And this is related mostly to the development of lung metastases as well as bone metastases. There are, importantly, extensive similarities between dog and human osteosarcoma. The genetics, the gene expression profile, the biology, and even the treatments are similar. Therefore, it is an important translational model where both species can benefit from findings that we get in this particular model. Importantly, in relation to that is also the fact that there are no reliable prognostic or predictive biomarkers for canine or human osteosarcoma. And here is where extracellular vesicles, we believe, may be important. What are extracellular vesicles? Well, they are membrane-bound vesicles that are released by cells into the microenvironment from where they can access biofluids, such as, for instance, blood, urine, milk, etc. So the route of release, the size, the markers that these vesicles express are going to vary depending on the type of vesicle, and the best known of these vesicles are exosomes. So exosomes are usually coming from the endocytic pathway, and they are produced by the exocytosis of the vesicles that are inside these multivesicular bodies. So exocytosis of these vesicles that are then the exosomes. So they are the best known and best characterized. These EVs are going to contain cargo, and this cargo is going to be DNA, RNA, proteins, or lipids. And this cargo reflects the cell of origin these vesicles are coming from, but more importantly also reflect the status, the biological processes that are going on in those cells, and they can be, when they are released, uptaken by neighboring cells, depending on where they have machinery to uptake them, and these cells can respond to this. So they are important for cell-cell communication as well. Importantly because they are reflecting of ongoing biological processes, they are considered nowadays as potential biomarkers of disease. And the way we envision this in the clinic is by, let's say, taking a blood sample and then putting this blood sample, the isolated plasma or serum from this blood, into a chip. And then this chip will be collecting these vesicles, binding to a particular marker they express, and looking at different target proteins that have been demonstrated to have prognostic or predictive value, and we can then quantify and use it as a biomarker of cancer. So this is the type of work we are trying to do, because if we want to use vesicles as a biomarker of cancer, we need to find out what those biomarkers are. And the way we are trying to find these biomarkers is in this macro project that we have developed here at Guelph that we have running for about 2 years, called Dog Bone. What we are doing in Dog Bone, we are a team of about 8 researchers at the time. We are basically joining of efforts and our expertise to be able to maximize the use of samples to accelerate the discovery of prognostic and predictive biomarkers of canine osteosarcoma. And the particular component I'm going to talk about today is the culture of explant, in this case from primary tumors, to isolate extracellular vesicles, EVs, and doing the protein profiling by mass spectrometry to identify potential biomarkers. So the methods are it's relatively straightforward. So we collect the samples, we separate the media from the media, we isolate vesicles, or what we believe contain vesicles, but then we characterize them to make sure that we have those vesicles we are looking for. And finally, we do mass spectrometry and analyze the results to see if we have any proteins that are potentially interesting. To go into more detail about the collection of the samples, so we have a canine patient that goes into surgery, and then following amputation, we collect sample of normal bone and tumor sample, okay? And then we mechanically dissociate it. We culture this tissue in the presence of media with serum, and this serum have been previously depleted from EVs so that they don't interfere with OEVs, and in the presence of serum for 24 hours. Then we collect, we concentrate the medium, and we separate it by size exclusion chromatography. And with size exclusion chromatography, we are going to get a number of fractions. And so this fraction then we need to analyze to identify markers of vesicles. In our particular case, there are 2 markers that have worked very well in dog lysates in our hands, and those are Flotilin-1 and CD63. So these are the markers that we use. So in this table that you have on the left-hand side, we have the A samples of osteosarcoma and A samples of normal bone. And as you can see, all of the A samples of osteosarcoma express flotilin, while only 6 out of these A samples express CD63. In the case of normal bone, only 4 samples express flotilin, and 3 out of these express CD63. And this is a representative Western blot showing this is a patient that we considered negative for normal bone for CD63, and you can see how the markers is seen in the different fractions that we collect. So what we do next is then we pull those fractions, and then we are going to analyze them by particle tracking. And particle tracking will tell us the concentration of different sizes of particles in our sample to figure out if we have the particles or vesicles we are looking for. And we also do electron microscopy, transmission electron microscopy. So this is the results of the particle tracking analysis. So you have normal bone and osteosarcoma, A samples of osteosarcoma. Each sample have a different color here that they are overlaid. And at the top, we have 4 samples of normal bone. As you can see, the diversity of the size of the particles in the osteosarcoma samples was a lot larger, and that is expected. I mean, it's a complex cell and heterogeneous cell population as well. And so you can see that the average particle size was about 145 nanometers in this case of osteosarcoma, and in the case of normal bone, about 200 nanometers. So we take these samples and we look at them on electron microscopy, and we observe vesicles there. A lot of the vesicles were in the 50 to 100 nanometers range of diameter the size. And you can see here in magnified that these vesicles were looking like a donut, which is a typical shape that you will see in these particles called exosomes. A lot of the particles were like this, but we also got particles that are a little bit larger, that are more compatible with things that are not exosomes as well. So this is not an exosome pure preparation, but that's okay because we didn't want to isolate only exosomes. We are interested in our vesicles. We just want to see if these vesicles can give us any information. So we did the mass spectrometry analysis, and using a number of softwares and engines, and we were able to identify a total number of 1,879 proteins. Of these, 355 appear to be only in osteosarcoma, 47 only in normal bone. Now, a lot of these proteins, we couldn't identify their function or what they were. However, for the ones we were able to identify, then they fell into different categories. And here, with these arrows in blue, I am trying to show you the ones that have the ones that belong to the larger categories. For example, a lot of them participate in cellular processes, biological regulation, metabolic processes, and response to stimulus. Interestingly, from a point of view, when we look at the proteins that show significant variation between normal bone and osteosarcoma, we were able to see that about 44% of those were proteins that were previously found to be in exosomes, which is a good sign because we know that we have something like exosomes in our vesicle preparation. So this is a very small list of some of the proteins we found. So for those proteins that were amenable for statistical analysis, we were able to find that 73 proteins were significantly different in normal bone versus osteosarcoma. And what I am showing you in this table are some of the proteins, a very limited number of the proteins that were enriched in osteosarcoma vesicles. And many of these proteins I am naming like PY and PX because of confidentiality reasons at this point. However, you notice that these ones that have asterisks are the real thing. So these are the proteins, RPS3 and RPS15-8, which are ribosomal proteins that have been previously found to be overexpressed in osteosarcoma in comparison to normal bone. And that is in humans, in human osteosarcoma. And apart from that, have been observed to have certain prognostic capability. But if you look at this carefully, you will notice that a lot of these proteins are part of the translation, protein translation, protein synthesis initiation. And in fact, what we went to see what pathways were enriched in our osteosarcoma versus normal bone. And this is, you can probably not see this name, but that's okay because I don't want you to see them. So, but what is here in purple is showing you, and this is the initiation of translation, of protein translation pathway, that I'm showing you is the ones that are in purple are the ones that were enriched in osteosarcoma EVs as compared to normal bone. So this is showing us that we have, for some reason, an increase of proteins that are especially involved in initiation of translation. This is an example of one of these proteins, and it was a ribosomal protein, and this is the volcano plot, and this, what you see in green here were things that were significant in osteosarcomas compared to normal bone, and this is one that is here, it's a ribosomal protein, one of my secret ribosomal proteins, And here you can see the samples individually for this protein, and you can see that the normal bone, many of the samples have none, only one has. And when you look at their matching partners, well, all of them have. And the number 8, normal bone number 8, and this one have higher as compared with the partner sample. So the protein, the ribosomal protein, one of the ribosomal proteins that was previously shown to be involved in human osteosarcoma is this ribosomal protein S3. And this is a study, and I particularly took this study because in this study, they only had 9 patients, but they have matching samples from normal bone and osteosarcoma from the same patient. And this is what I am showing here. So this is their immunohistochemistry showing that in normal bone, the expression was very, very low, and in osteosarcoma, very high. When they quantified the expression, they observed that the patients that had long metastasis and diagnosis have higher expression as compared to the patient that did not. And then when they did the analysis of survival over time, they observed that the patients that have higher expression, in fact, have worse survival than the ones that have low expression, although it was not significant, but this is a very reduced number of patients, as you can see. So you wouldn't find significance in this small population. So in summary, we have found that we can identify floodling and CD63 EVs, and we can obtain them from explants of osteosarcoma and normal bone. And these EVs include, and they are not only exosomes. And this is suggested by particle tracking analysis, electron microscopy, and the proteomics as well. We identify different protein cargo in the normal bone versus osteosarcoma. Interestingly, osteosarcoma appears to be enriching proteins that are involved in translation, and some of these are ribosomal proteins that indeed were shown to have a role or appear to be involved as well in human osteosarcoma. So our future work now, well, we need to validate all of this data with antibodies in tissue and so on and so forth. We also need to include a larger number of samples because we only have 4 normal bone here and we need more. But we are also including EVs from metastatic nodule explants that we think are going to bring even more interesting results in terms of how the tumor progresses and what the EVs can tell us about metastasis. So I just want to remind you what is the significance of this work. Well, apart from we started in thinking, okay, this is going to lead us to prognostic or predictive factors for canine osteosarcoma. But some of these proteins, and I already started looking at the ones we had, are potentially targetable. So we may even get more efficient treatments for canine osteosarcoma. And because canine and human osteosarcoma are so similar, especially canine and pediatric osteosarcoma in terms of biology, gene expression, and so on and so forth, we can probably translate the results to humans and vice versa, get some important things from humans that can be applied to dogs. Finally, I would like to acknowledge the people that made this work possible. This looks simple here, but this has taken us 2 years to get where we are here. And I want to thank, in particular, clinical trial coordinator Vicky Sabine and the tumor bank coordinator Kaya, who is here today, because without their help, we couldn't have implemented this. The patients, of course, that have given us their tissue, thanks to the donors that signed the consent for us to get this, and the people that have collaborated with us for the proteomic analysis, in particular, Dr. Janusz Rack and his postdoc, Don Sik Choi in McGill University, and Lauren Taylor, which is the manager of the facility at McGill University. This work was funded by NSERC. All of the optimization for the protocol for the culture of explants was funded by NSERC, and the actual proteomics and all of the analysis by Petros. I want to give a big thanks to my dog bone team, some of who are here today, because this is also thanks to them that this project has been possible, both conceptually and logistically. Thank you, and we'll receive your questions.
Time for maybe one question.
Yes.
It's really cool work. And this whole area of extracellular vesicles is really quite interesting, especially looking at cytokine levels and circulating. And that's the question you alluded to it in your significance specific area. But if this truly is going to be a biomarker, we want to be able to measure it in the peripheral blood compartment, right? And so have you started looking for these things in your samples of your dogs? That way you don't have to sample them, biopsy them. It's hard to get metastatic samples in a living dog.
Yeah, so the whole idea, I don't know if you look at the slide that we have for the dog bone project, so let's look at it more carefully. So this is the dog bone project here. So basically what we are is collecting blood samples and then collecting these tumor samples and kind of simultaneously, okay? So these blood samples we take at pre-treatment, we also take during treatment, okay? And we have plasma from all of those patients.
It has to be.
Yes, we are starting to isolate them from the plasma.
Yeah, the plasma is really difficult, but it can be done.
Oh yes, we are.
You can't just like collect regular plasma per se.
We have isolated them from plasma, and we are now improving this method with the help of the people at McGill, who is actually doing this on human patients. So yes, we are doing that, and what we are planning to do in the case of the information that we get from these explants is to see if we can bring this information to the plasma. And this is very interesting because I show you these signatures that we are getting, right? And there is a group in China now that have done the same in plasma and in getting very similar signatures, and this is with human patients. So we are optimistic that we could potentially get there.
Start right into it. This is Louis. Louis is a 6-year-old male neutered English bulldog who was undergoing treatment for lymphoma with the CHOP protocol. And once the prednisone was off, the owners wanted to put him back on his NSAID because of osteoarthritis. So went back and looked at his biochemistry, that pre-treatment biochemistry, to make sure that renal function was okay. And his creatinine was fine. That was okay, but his SDMA was very elevated. So SDMA, which is symmetric dimethyl arginine, is a biomarker of kidney function. Like creatinine, it's cleared exclusively by the kidneys, but unlike creatinine that comes strictly from muscle, SDMA comes from all cells. And for that reason, when animals have muscle wasting, is a better biomarker of kidney function than creatinine. So it was elevated, which is odd, because most dogs, they're proportional. And then the next day, in comes Honey. So this dog, when he's in complete remission, now the dog is in complete remission, so went back and remeasured SDMA on the dog, and now it was down to 8, from over 20 down to 8, which was a significant drop. There's some biological variation in this marker, but not a lot. Then came in Honey the next day. She's a 10-year-old pug. She had naive lymphoma. Her creatinine was 122, so just in the upper end of reference range, and her SDMA was also elevated. She then, on the second week of the CHOP protocol, was judged to be in complete remission. Her creatinine had dropped, indicating that on the first day, some of that elevation in creatinine was prerenal, but SDMA had dropped as well. All right, so the question is then, we're seeing a apparently disproportionate elevation in creatinine, in SDMA compared to creatinine. To really truly say that, though, we have to have ratios. And ratios haven't really been as much discussed as absolute values. But if we go back in the literature, for dogs with acute kidney injury, they had a median value of SDMA of 6.5, but all the way up to 20.9, which makes sense in acute kidney injury that you would have some elevation in the ratios. So these are ratios. And with dogs with chronic kidney disease, where SDMA is first marketed, the overall ratio is 10 with some elevation. So in the case of Louis, the SDMA ratio dropped from 29, about 21 down to 6.7, so normal. And for Honey, it dropped from around 16 down to 12. And just to give an idea, if we were to take an average normal SDMA in a dog, it'd be around 9, and creatinine in around mid-reference range to slightly low end of reference range would be 78, and a typical ratio would be 10. On rest of workup, Louis had some small nodules in both kidneys that could have been infiltration, and sure enough, when the dog was in remission, those had resolved, so there probably was some renal infiltration. Honey had not had ultrasound as part of staging, but the kidneys were normal once in complete remission. All right, so based on that, we went back and looked at other cases, both here and at the cooperating referral practice, veterinary 404. And of all those cases together, 25 active cases and 7 inactive ones, looked and at pre-treatment SDMA, and it was elevated in well over 50%, close to 70% in both hospitals. So based on that, then we started to recruit new cases with naive lymphoma, ensuring that that every time they came in, SDMA would be measured so we could track them throughout the progress of disease. And as well, because abdominal ultrasound is semi-elective, it's encouraged, but doesn't occur in every case, it was put in there and funded so that we could have it all the time, pre-treatment and during remission. So based on that, and of course there was informed owner consent. So this is the result. We can see that this is based on paired and unpaired linear regression. If we look at dogs with naive lymphoma, 73% of them had elevated SDMA, which was significantly lower when they were in complete remission. And these are looking at the ratios, which do the same thing. So in those dogs, 35 of 40 dogs had an abdominal ultrasound, and only one of them had evidence of infiltrative disease, suggesting that most of them did not. Now, ultrasound is a good diagnostic tool, but there's no doubt that it can't pick up very low-level infiltration. So it'll pick up only to a certain level. So it's still possible that there is infiltrative disease in these. So based on naive dogs, so looking at these naive dogs, all of them had aggressive multicentral nodic lymphoma. They all had typical signalments. 40% were immunophenotyped, and without showing you all the statistics, essentially there was no correlation with any typical parameter used to characterize lymphoma in dogs and SDMA level. All right, now that is, so if we kept following Louis, Louis unfortunately then relapsed, and this is his SDMA. So when he was naive, he was 22, in remission 8, in complete remission, judged to be right at the end of the protocol. It's beginning to go up, which didn't make us raise a weary eyebrow about this. And in relapse was 19, and then when he had progressive disease, 23. So it followed his bulky disease very closely. So if we then go and look at all other dogs, comparing remission, naive lymphoma versus relapse, see that there's essentially no difference. So bulky disease, whether before treatment or after treatment, had the same results. All right, and then if we looked at the dogs that were trending throughout treatment, when they were in partial remission, they were intermediate values as to be expected. Also, there were several dogs with extranodal lymphoma, 2 dogs with renal, 2 dogs with GI, and 1 dog with acute lymphoblastic leukemia, and essentially the results were exactly the same as with nodal lymphoma. So the conclusion is over 50%, that's a conservative statement, it's probably more like 70%, of dogs with aggressive lymphoma have a disproportionate increased SDMA compared to creatinine. All right? When those dogs achieve complete remission, the SDMA normalizes, and when they relapse, it goes back up. So why is this? Now, first of all, I have this as preliminary because, this data is not mature. There's like 1,000 more samples sitting in a freezer and lots more clinical data, but all it's going to do is increase the power. So what are the features here of lymphoma? So it's an acute compared to other neoplasms. It's an acute tumor. Because it's acute, muscle mass is normal, so that's not going to be affecting creatinine versus SDMA, unlike a dog with a chronic tumor, where if there's a lot of muscle wasting, then there'll be a disproportionate reason for that. But because SDMA doesn't have that, we'd expect them to be proportionate. There's a very high metabolic rate, and since SDMA is produced by metabolism, maybe that is relevant. It also is a tumor that can have spontaneous tumor lysis. It varies a wide age range of dogs, so a lot will not have chronic kidney disease. And I would think it's safe to say that most dogs being treated with lymphoma, there's not a usual concern about concurrent kidney disease. All right, so if we look at these features and look at possible mechanisms for elevated SDMA, so it's easy to think that SDMA could go up because of metabolic rate. If we look at human lymphoma, though, ADMA, which is asymmetric dimethylarginine, but not SDMA, is elevated. And then Yaramilli, this investigator here, is the scientist at IDEX that developed SDMA as a test in dogs. Based on some preliminary results, him says it's the same thing in dogs. ADMA will go up, but not SDMA. All right. So based on that, the implication that, at least in most tumors, that SDMA actually reflects a decrease in kidney function. Now, the other possibility, though, is just to see if if anything else would fit with that. The other main classic marker of metabolic rate would be in lymphoma would be lactate dehydrogenase. So on remaining serum samples, that was added on for like 161 samples. That did not correlate with SDMA. We also have to keep in mind in trying to analyze this data that LDH is also cleared by the kidney. So it's hard to find something else that's not affected by renal function. All right, what about tumor lysis? So metabolic rate, probably not. What about tumor lysis? We know that tumor lysis syndrome causes acute kidney injury. Is it possible that this is low-grade tumor lysis causing low-grade kidney injury, enough to be reflected in SDMA, but not in clinical kidney disease? Based on that, potassium was normal in all dogs. If we look at normal markers of tumor lysis, a low number of dogs did have hyperphosphatemia and a low number had hypocalcemia, but not nearly enough to account for the number of dogs with elevated SDMA. Just like LDH was added on as another marker of tumor lysis, went on to bank samples and added on uric acid. There was, and if we look at naive dogs, all of them had the average, although they overlap, the average uric acid level was higher than dogs in complete remission. And then relapse went up. So this pattern of uric acid starting high, dropping, going back up did more or less parallel SDMA, so tumor lysis may be a possibility. The other problem, though, is uric acid is also cleared by the kidney, so it's hard to get renal clearance out of there. And it wasn't particularly well coordinated with creatinine, but as we would expect, it was moderately correlated with SDMA. And so that brings up the question, maybe SDMA is simply a more sensitive marker for tumor lysis than, or for uric acid nephropathy than any of the other traditional markers. All right, and the third mechanism, which is the one proposed by Yaramilli, is the AGXT2 enzyme is responsible for clearing ADMA, SDMA, Yaramilli has reported that the enzyme sitting on the basement membrane of the kidney is got altered polarity, and that altered polarity is due to beta-aminoisobutyrate. Now, beta-aminoisobutyrate is a product of pyrimidine catabolism, and it's also cleared by this enzyme, the same one that clears that. And so there's a possibility that although this is a normal enzyme-substrate interaction, that somehow elevated levels of BAIB is somehow altering the enzyme so it affects its other constitutive function. And in particular, since there appears to be altered basement membrane polarity, is the fact that creatinine has a neutral charge and SDMA is a strong cation. So possibly breakdown products of pyrimidines are altering the enzyme such that SDMA is elevated. Now, the other thing this enzyme does, it is responsible for the metabolism of glyoxylate down to glycine. And if that is impaired somehow, it could be actually pushing it to the other metabolic pathway and produce oxalates. And oxalate nephropathy is a well-known problem in small animal medicine because of ethylene glycol poisoning. And it's possible then that maybe BAB is somehow pushing this into oxalates, and oxalates are causing decreased kidney function. However, we also have to keep in mind that AGXT2 has polymorphisms in human medicine, and it's possible that just simply polymorphisms in dogs, and the dogs with one set of enzyme, with one enzyme form can raise SDMA and the other can't. So maybe it's strictly that. All right, so, and the last mechanism is that this is actually tumor infiltration into the kidney that's occurring below the level of ultrasound detection. Unfortunately, for clinical patients, there's no other practical way to go after it. There are some enhanced ultrasound techniques that could be done. Scheduling is still always a problem with patients coming in and out and trying to get the ultrasounds done on them. Biopsy is certainly not going to happen. And there could be more attention at necropsy to dogs in relapse to look for kidney infiltration. All right, so regardless of this, I think I skipped over a slide here. Yeah. Okay, so regardless of the mechanism, though, the key thing is if this is an acute kidney injury, it's a reversible injury. So what does this all mean? Is there any clinical relevance? It's very interesting, but so From an internist perspective, the occasional case is actually presented for an elevated SDMA for us to investigate kidney function, whereupon we just find lymphoma. That has happened in a couple of cases. As a biomarker of kidney function, it's important for a clinician not to diagnose typical acute kidney injury or chronic kidney disease and use that as a reason not to treat the dog for lymphoma. For example, say, well, you know, I'm sorry your dog's got lymphoma, we could treat it, but hey, the kidneys are sick, so we won't bother. We have to make sure that doesn't happen. But beyond that, and clinicians, please feel free to disagree with me, overall, kidney diseases aren't that big a deal in dogs with lymphoma. So from the oncologist perspective, this is to some extent a biomarker, maybe a biomarker of tumor, and maybe a biomarker of response, because it is correlating, at least in some dogs, to tumor burden. But we have to be careful not to flip this on its head, because we're judging SDMA validity based on a clinical judgment of remission, which still remains the standard. So is it not necessarily valid to flip that around and say, oh, well, then SDMA, we're going to use that to judge remission? People are always looking for ways to evaluate minimal residual disease. Could this be one more mechanism? Possibly, but given some of the sophisticated molecular tools that are out there, this is probably way too crude. Maybe, as people are always coming up with predictive models, putting so many parameters into it, maybe this would have more utility in conjunction with other biomarkers. There's always an attempt to characterize subsets of lymphoma, hopefully with an implication that treatment could change. Maybe this would help characterize lymphoma subtypes, in which case then maybe it'd have a role in precision medicine. At this point, though, it really remains an observation, brings up some interesting possible mechanisms, and of course we never know exactly how that'll play out. Okay, I'd like to acknowledge basically everyone at the Animal Cancer Center, including the dogs, of course, Dr. Binsley in Pathobiology, Dr. Zerlinden reviewing ultrasounds and diagnostic imaging, clinicians at VCA 404, and Pet Trust funding, as well as some private donations.
Thanks, Tony. Got time for one question.
Sorry?
Has anyone measured ADMA?
Yaramilli has, but he's not reported it. It's just been in a statement in the poster and in the oral presentation that ADMA is elevated, but beyond that. And because I was being so careful not to talk to IDEX about this, not to try to, you know, but just because I wanted to declare no conflict of interest, really not discussed it more beyond that. But I've not seen any new data on ADMA. But the test to measure it does exist.
Are there any questions? Thanks, Tony. So I'll just introduce Dr. Pizatusi from all the way here from University of Saskatchewan in Saskatoon. There's a vet college there, in case some of you don't know that. Most of you probably will. And they're starting up a new oncology sort of group there. And we're going to get a bit of introduction about that, but also your own research. So.
Yeah, thank you so much. Good afternoon. I'm very excited to be here, and a big thank to the organizing team of this wonderful symposium to give me the opportunity to come here and share some of the data and information with you. Today I'm going to talk about F receptor tyrosine kinases and their potential for development of targeted cancer therapies for both humans and companion animals. So I divided my talk today in 3 sections. The first section I'm going to talk about some research data, these data coming from human cancer research side, which I spent most of my previous time in that area, and then I'm going to talk about comparative oncology and how those experiences and research data could be utilized in a comparative oncology approach. And also I'm talking about some other research at the University of Saskatchewan for seeking potential collaborations. So I had the opportunity to work with different models of human cancer research, including leukemia, colon cancer, prostate cancer, and breast cancer. For today, I'm going just to focus on the work on breast cancer because it has more relevance or clinical relevance in terms of using DNA damaging drugs, the small molecule inhibitors, and synthetic antibodies. So let me introduce the F receptor tyrosine kinases. F receptor tyrosine kinases are the biggest subgroup of receptor tyrosine kinases. They are divided into two groups, FA and FB. There are 9 receptors in FA group and 5 in FB group. They are expressed at cell membrane. The difference between FA and FB groups are the type of ligands that they bind to. FA receptors, they bind to ephrin A ligands, and FB receptors, they bind to ephrin B ligands. Both kind of ligands, they are expressed at cell membrane too, and therefore F receptors are very critical for regulating cell-cell interactions. So why F receptor tyrosine kinases are important for in cancer research? If you look at their function, they regulate cell proliferation, morphology, adhesion, movement, survival, and differentiation. These are all important functions that either being altered or needed by cancer cells. And indeed, abnormal alterations of F receptors have been detected in multiple human malignancies. So when we talk about alteration, what type of alteration are we talking about? I'm going to talk about 2 main conditions. Either we have decreased expression or function of receptor, or we have increased expression or function of a receptor. And I'm going to talk about 3 different scenarios, how to deal with this condition to have some suggestions for clinical implications. In the first case, when we have decreased expression, if that decrease is partial, we can plan therapy based on the function, based on the remaining function of the F receptors, or even by enhancing that function. And if the decrease is complete, or that reduction is complete, it's a complete elimination of the receptor, we can detect lethal pairs with that target molecule, which I'm going to expand on that. And in the case that we have increased expression or function, we can design synthetic antibodies to interfere with those cancer-promoting functions of the receptor. So let's start with the first scenario. Expression of F receptors is increased. Most of the time, they are increased in multiple human malignancies. There is one receptor, FP6, which its expression is consistently reduced compared to the normal tissue. So that raises the question that what aspects of function of FP6 is not desirable or not welcome by the tumor, so they want to get rid of it. We try to answer that question. As I mentioned, in general, F receptor in FB receptor expression is reduced in cancer compared to normal tissue. However, if you look at this panel, the FP6 receptor expression is better preserved in triple-negative breast cancer tumors. So there's still some function remaining in triple-negative breast cancer compared to other subtypes of breast cancer. So we built our model, research models on triple-negative breast cancer. The cell lines that we used for our investigations were MDAMB231. In this cell line, FP6 promoter is methylated and there's no expression of FP6. So we restored FP6 expression in this cell line. BT20 and HCC70, they have endogenous expression of FP6, so we use shRNA to silence FP6 expression. So for our experimentation, basically we have cells that either express or have FP6 or they don't have it. So one of our first findings was that FP6 increases cell-cell contacts. These are MDA231 cells. You see 2 colonies here. Each of these colonies developed or grew from a single cell. So in control cells, you see how the colony looks like. Their cells are elongated. They invade to the surrounding of the colony. There are lots of space between the cells. However, when we restored FP6, that increased cell-cell contacts and they form these kind of closed colonies. We also test motility of the cells in regards to FP6 presence, and we found that FP6 reduces cell motility. This is scratch or wound healing assay. So basically, it's a monolayer of cells. We make this scratch, and we monitor how fast cell can come into the scratch and close it. And you see after restoration of FP6, even at 36 hours, there is still significant gap left compared to control, which was, the gap was completely closed. So if FP6 increases cell-cell contacts and reduces cell motility, can it reduce tumor invasiveness? So we wanted to test that. We injected MDA cells with or without FP6 into mouse. We found very curious observation there. If you could look at here, the tumors that had restored FP6, they had greater growth rate and faster development compared to the controls, especially at the initial time points after injection. So what could cause that? Why those tumors are growing faster? We look at the angiogenesis in the tumor, there wasn't any difference in blood vessel formation in those tumors. There was another possible answer for that, which I'm going to talk about it. So, you know, if we culture cells, or even when we isolate cells from a tumor, the population that we are dealing with is a heterogeneous population. And it means that not all type of cells that we have in population are able or capable of developing tumor when injected into a mice. There is basically, or in fact, there is just a little or small subpopulation of cells that are capable of developing a new tumor. We call them tumor initiating cells. So we are seeing that increased growth rate, initial growth rate, when FP6 is restored. Could that be due to increased proliferation of tumor initiating cells? To test that, we cultured these cells in the presence of FP6 or absence of FP6 in tumor spheres. Tumor sphere is a specific culture condition that favors only growth of tumor initiating cells. And by doing that, we observed that yes, indeed, FP6 increased proliferation of tumor initiating cells. This is very interesting because tumor initiating cells usually stay in a dormant, less proliferative state. That's one of the reasons they are responsible for drug resistance, especially when DNA damaging drugs are used, like doxorubicin. So because they are not proliferating very fast, they're still there and they can cause tumor relapse. So if FP6 kind of pushes them out of their dormancy or less low proliferative state, can it make them more sensitive to DNA damaging drugs? So we tested this hypothesis in animal model. Here is a PDX model that we used. And you see tumors that had FP6, they had higher growth rates. They developed faster or bigger. However, they had more profound sensitivity to doxorubicin compared to their controls. In fact, if you look at this graph, in the presence of FP6, we had about 80% reduction in tumor size when tumors treated with doxorubicin. And in control, we had only 10%. So what's the clinical message from here? We could recommend that DNA damaging drugs are better choices for treating tumors that have higher levels of FP6 expression. So let's see and talk about this next scenario when we have complete elimination of a receptor. So we need to find synthetic lethal pairs in that situation. But what is that? So there is a concept of synthetic lethality. The idea is that the function of two genes are somehow related and they are necessary for cell survival. When both of them are intact, the cells are in good shape. And when one of the genes are eliminated or suppressed, the other gene can compensate for the lack of that gene. And again, there is no adverse effect on the cells. However, when both of them are eliminated or suppressed, then that is a lethal combination for the cells. So in tumors that eliminate FP6 for any reason that they don't want it, we can find what other genes are lethal pairs with FP6 and then try to target those in order to have a lethal effect on cancer cells. So we did a screening to find those lethal pairs with FP6. It needs a condition of FP6 plus or minus, and then we silenced up to 18,000 different genes individually in both conditions. we do high throughput screening, and from that, we found actually 113 potential lethal pairs with FP6. It means that in the condition that FP6 is absent, suppression or inhibition either of these genes individually can be lethal for the cells. So we narrow it down further. We did some further analysis. You see here functional network analysis. We found Src kinase as a central molecule among those 113 other factors. So basically, our research shows that in tumors that eliminate FP6, if we use Src kinase inhibitors, such as KX2391, we should see more profound effect on the cells in terms of inhibition compared to the condition that FP6 is present. And we did that experiment. We treated cells without FP6 or with FP6 with Src kinase inhibitor, and as we hypothesized, we had bigger inhibition when FP6 was absent. So what is the clinical message from this experiment or this project? We found that FP6 and Src are a synthetic lethal pair. So this means for tumors that don't have FP6, Src inhibitors could be a good or possible option for treatment. However, in tumors that maintain FP6, the use of Src kinase inhibitors may not be very effective because the remaining function of FP6 can compensate for that inhibition. So KX2 actually is a drug, FDA-approved drug, which is in clinical trials for breast cancer and prostate cancer. But however, there are very diverse responses from individuals being treated with this drug. So we could see that some other genes could be responsible for that resistance development. And last, when we have increased expression or function of F receptors, we can target that with synthetic antibodies. This project has been done with our industry partner Biomedical. Unfortunately, due to their request, I cannot disclose the name of antibody or its target molecule. But you can see that we injected MDA MB231 cells into mouse, and upon tumor development, we started treating them with synthetic antibody or IgG control, and you see there was huge and profound inhibition on tumor development when we used synthetic antibodies against one of the members of F receptors. So with this, I'm going to talk a little bit about how these data and experiences could be used in a comparative oncology approach or for companion animal cancer research. Well, F receptor tyrosine kinases are gaining a lot of attention in human cancer research. And look at this list of possible similar malignancies between companion animals and humans. Investigating the role of F receptors in companion animal malignancies would be very promising. It definitely has benefits to both animals and humans. It could increase collaborations and opportunities for getting research funding and also encourage industry contributions. So this is the area that my lab is focused on right now to basically investigate the role of F receptor tyrosine kinases on companion animal malignancies. This is the optimum, workflow. However, if you look at here, we already know some valuable and promising targets from human cancer research, which we could use them in terms of synthetic antibodies or small molecule inhibitors in the context of companion animal cancer research, which is very valuable. So with that, I'm going to the third part of my talk and give you some idea of University of Saskatchewan and the research that is going on there. I'm starting with our Western College of Veterinary Medicine. We are actually the first vet college in Western Canada, including Saskatchewan, Alberta, and British Columbia. We are developing the oncology center there, which provides medical and radiation oncology services, and we are also developing comparative oncology research group there. And we have this allotrusin imaging suite that opened few months ago, and we have a full-size PET CT scanner then that is going to be used for both clinical and research purposes. And as a matter of fact, they started using that a few weeks ago, actually. And we have also a rehab center in our future oncology center. There are 4 phases, 2 phases have been already completed. And another news is that we are another Canadian member of Comparative Oncology Trials Consortium as of a few months ago. So about College of Medicine, we have translational cancer research cluster there. Some of the data I showed you with high throughput screening or synthetic lethality we did in collaboration with College of Medicine. We have also Saskatchewan Therapeutic Antibody Resource. They have developed a new synthetic antibody library, which is very valuable for our research and translational work. We have also facilities for genomics and proteomics analysis in College of Medicine. And last but not least, we have Saskatchewan Cancer Center for Cyclotron Sciences. So actually, one of the antibodies that I talked to you is being radio-labeled at this center for use, diagnostic use, or at least research for PET-CT imaging. So it's not very clear, but it's a big view of our campus. The point that I wanted to mention here, do you remember this building? Okay. So this is our vet college. The oncology center would be on the west side of it. And here we have the cyclotron, and here is the College of Medicine, Health Sciences, and Medical Hospital, Veterinary Hospital. My point is all these centers are within walking distance from each other and really encourages collaboration between all these centers. And as a researcher in oncology in vet college, I would be very happy to facilitate any collaborations between our colleges and our universities. And thank you so much for your attention.
Thanks. Any questions?
So some of the F receptors are also called angiogenesis. And I know that you showed that there wasn't a difference in angiogenesis between your aggressive and non-aggressive tumors, but when you're using these inhibitors, are they going to be targeting, like, is it a dual target? Are you targeting the neoplastic cells and also perhaps blood vessels? Do you know that?
I'm sorry, is your question about the antibodies or small molecule inhibitors? Yeah. Like that one that we investigated about the new angiogenesis was specifically about the FP6 receptor that wasn't involved in new tumor.
Cells, right? But when you treat a tumor with your inhibitors, whether they're large molecule antibodies or small molecule inhibitors, are you also going to be inducing, in addition to an anti-neoplastic cell effect, an anti-angiogenic effect? Have you looked at that?
We haven't looked at that. It's just for inhibitors, again, specifically, we try to use the FDA-approved inhibitors, which are already available. So they have already done some of the testing in terms of toxicity and some other side effects. But for antibodies, synthetic antibodies, definitely that's something we need to look into it in more detail. Yes?
So we do have some, as you know, commercially available certain inhibitors in this thing, which have been used in leukemia, and there are published PK data on that.
Right.
So that would be an easy add-on, an off-the-shelf type of thing. And since we see much more sarcomas in our canine population than in our human population, albeit for the pediatric cancers, I mean, there's some great stuff here that could be applied.
Exactly.
Surrogate.
Yes, no, I agree with you. And it was amazing even with our work, which are not, it was a small part of it. We saw that there are lots of opportunities to develop personalized type of treatment, even for each.
So have you looked at any of the osteosarcoma cases or angiosarcoma cases for loss of your ERV6?
Not yet. It's just, I started from January, but it's just trying to develop. So, but that's my on my list. So we completed designing those, like whatever to setting up the assays to look for expression of all these F receptors in different malignancies, canine malignancies. Yes?
So I'm just looking at the phenotype that you have with the cells, so you get increasing cell-cell contact.
Yes?
By
yeah.
increasing the levels of the 6.
Yes, that's true.
And so, and in triple negative, what is can you go back to
That cell line that I showed you, they completely eliminate FD6. Like the promoter is methylated and no expression of FD6.
Okay. So, but when, and then in the cell, when you re-express it, then you increase cells.
Yes. There. Sorry, go ahead.
So why is it happening? Is it that you are regaining in-cadherin or any other cadherin?
Yes, it's a process. It's called epithelial to mesenchymal transition. So I didn't have time to show all the data for that, but it's a big part of our work that is published in Oncogene. FD6 actually increases some of those epithelial markers after we restore that. It significantly reduces beta-catenin transcription in the cells and function. And through all of that, it's also physically causes those cell-cell interaction. We did like localization experiments. We saw that FD6 is located at cell-cell contacts area. We stained them with ZO1 and FD6, and they were overlapping. But it's more complicated than that. It's just the physical cell-cell, increasing cell-cell connection. It's reversing that epithelial to mesenchymal transition, which is very interesting. Yes?
One more follow-up question. So in the dogs, so in the models that you showed where FB6, if you, so you'll get more tumor mass, your growth rates will go up. What about metastatic rates? Intuitively, I would think it would fall off.
Yes, the metastasis, we started to evaluate that. We haven't completed that in that project, but that was on our list. We did some preliminary data. We saw at least microscopically, by counting those metastatic areas, that was a reduction in metastasis. So, but
more tumor primary, but less metastatic.
Exactly. Yeah. Yes. No, exactly, because it reverses epithelial to mesenchymal transition. So that process is needed for a tumor invasion and metastasis. However, the effect on the ticks or tumor initiating cell is increasing their proliferation. It's to their disadvantage, because they become more sensitive to
so we can have Michelle have primaries and you're all the...
Okay, thank you very much.
Thank you so much.
And our first speaker is Matthew Chang from the Department of Food Sciences.
Yes. Good afternoon. My name is Matt. I'll be presenting to you some of the major highlights of my PhD project. So, acute myeloid leukemia is this devastating blood cancer that starts with the HSC population shown here. And their role is to produce our body's functional erythrocytes, granulocytes, and thrombocytes. Occasionally, members of the HSC pool can undergo a leukemic transformation to form a leukemic population. At the very apex of this population are the leukemic initiating cells, shown here as the SLICs, and their role is to produce the bulk of the leukemic population, known as the clonogenic blast cells. For the past 40 years, we've been basically reliant on 3+7 induction therapy as our frontline anti-AML regimen. Unfortunately, remission rates tend to vary and also decrease with the more mature patient population. This is primarily because of treatment-related comorbidities and the fact that these compounds just weren't designed to target that initiating cell population that's known for relapse. Recently in our field, several papers have highlighted that fatty acid oxidation is critical to leukemic survival, proliferation, the initiation of relapse, and even the maintenance of chemoresistance. And it's this pathway that's the main method for most normal and malignant cells to break down fats that are less than 16 carbons in length. This pathway starts with fats entering the mitochondria via the CPT system, carnitine palmitoyl transferase system. Once in the mitochondria, the fats are then broken down by different sets of enzymes depending on how long that initial fat was. The end result of one round of fatty acid oxidation is the production of acetyl-CoA for the TCA cycle, NADH for complex 1, and FADH2 for the electron transfer flavor protein. And it's because of that literature I just outlined on the previous slide that inhibitors of this pathway are now being considered a possible chemotherapeutic candidates for AML. My work has been centered around this mixture known as Avocan B. It's a 1-to-1 mixture of two 17-carbon long fatty alcohols, complete with hydroxyl groups at the 1, 2, and 4 positions, and either a terminal double or triple bond. In my early experiments, highlighted here in red, I showed that avocadine, that triply bonded fatty alcohol, was the most potent component of this mixture in both leukemic cell lines and also primary patient samples, all the while sparing the clonogenic growth of the healthy hematopoietic population. We also evaluated the in vivo efficacy of this compound, avocadine, and showed that it significantly reduced primary and secondary engraftment in a patient-derived xenograft study. We also wanted to study the effects of the avocadine compound on mitochondrial respiration. So we turned to high-resolution respirometry. This is an example of one of these experiments shown here. So across the x-axis, you have experiment time going from left to right, and on the y-axis, on the right-hand side, you have oxygen flux represented by the red and gray traces. So after a brief treatment with our compound, we injected intact cells at this point, and that increase in flux that you see there to that plateau is respiration that is coupled to ATP synthesis. It's powered by whatever substrates the cell had access to in cell media. We can also look at maximal respiration over here with an injection of oligomycin and then FCCP. And then lastly, it's customary to end off the experiment with some sort of selective OXPHOS inhibitor. In this case, I chose antimycin A to basically bring respiration back down to 0 and prove to you that all the flux you're seeing here is due to the mitochondrial electron transport chain. And the arrows shown up there are basically what I quantified in these bar graphs. And in short, we once again saw that avocadine, that triply bonded fatty alcohol, was the most potent component of this compound in terms of inhibiting leukemic mitochondrial respiration. And because of that, we'll be carrying on with just this compound for the rest of our experiments. We had this hypothesis, though, that this compound wasn't actually inhibiting the electron transport chain. It was actually inhibiting fatty acid oxidation, which lies upstream to OXPHOS. So we had to modify our protocol. You can't really study fatty acid oxidation with whole cells because even if you supply fat to a cell in media, there's no guarantee that your cells are actually using fat exclusively to power the electron transport chain. So to get around this, we had to permeabilize our cells prior to injection, and what this does is it basically strips away all the cytosolic substrates inside the cell that's powering OXPHOS, while leaving all the enzymes and mitochondria and other organelles intact and functional. That's why there's cells in the respirometer from this point to this point, but there's no respiration. It's not until I inject palmitoylcarnitine, malate, ADP, this cocktail, that we actually see an increase in oxygen flux, which we can exclusively say is due to fatty acid supported respiration, because fat is the only substrate that's in the cell, inside the respirometer with the cells at this moment. So once again, the arrows are basically what I quantified, and shown down here. In short, we were able to show that this compound inhibits fatty acid supported mitochondrial respiration. We weren't ready to call this compound a flat out fatty acid oxidation inhibitor just yet. We wanted a more direct measure of fatty acid oxidation, and this led us to a collaboration with Dr. Akhtar from MCB. He kindly ran these experiments for us. We basically supplied a radioactive palmitate to our cancer cells. This gets broken down to radioactive acetyl-CoA, which can then enter the TCA cycle just like normal. Eventually, this acetyl-CoA is liberated as radioactive carbon dioxide. And that release of CO2 is our measure, a more direct measure of fatty acid oxidation. This is more direct compared to respirometry, because respirometry kind of ties FAO activity to oxygen consumption at the electron transport chain. Anyways, we were able to show that this compound actually inhibits fatty acid oxidation in both of our leukemic cell lines. The next question I had was, okay, great, we have this compound that we are pretty sure is inhibiting fatty acid oxidation, but which one of the enzymes here could it be interacting with? What is the molecular target? This led us to another collaboration done in conjunction with some researchers out of the University of Pittsburgh. They specialize in the study of fatty acid oxidation deficiencies in humans, and this experiment is known as acylcarnitine profiling. The gist of this is that whenever you have some sort of deficient activity in any one of these enzymes, there's going to be a massive increase in accumulation of its substrate inside the mitochondria. In an attempt to alleviate this inhibition, the mitochondria will try and have some of this substrate converted to a acylcarnitine intermediate, which can then get pumped out of the mitochondria and eventually out of the cell. This can then be identified and quantified using mass spectrometry. So in our collaborators' field of study, this is commonly done and is a clinically relevant procedure. When a patient comes in with like any one of these FAO deficiencies, they basically are using mass spec to look at what accumulations or depletions in specific metabolites to diagnose whether long, medium, or short chain fatty acid oxidation is being inhibited, and possibly even down to which exact enzyme. So we wanted to apply this same methodology to our project. We used their cells treated with our compound, and basically over here on this slide, WT wild type stands for one of their fibroblast lines that was derived from a healthy donor with functional fatty acid oxidation. We treated this cell line with either a vehicle control or our compound of interest. In addition, we had a positive control for fatty acid oxidation in the sense of a VLCAD deficient line. VLCAD is one of the enzymes associated with long chain fatty acid oxidation. So when we looked at the results, there were over 30 fatty acid intermediates highlighted and identified in this mass spectrometry analysis. Only 5 of them were shown to be significantly different from control to treated groups. So in our discussions with our collaborators, they were telling us, hey, you know, this accumulation of C16 that you have in your cells indicates to you that most likely there's some block at long chain fatty acid oxidation. These enzymes are the ones that process palmitoylcarnitine and fats of that length. In addition, this significant depletion of acylcarnitines from C10 through to C4 is indicating to us that, you know what, the medium and short chain fatty acid oxidation enzymes are actually probably fully functional. They're not affected by our compound. And that's because this blockage early on in the fatty acid oxidation pathway is preventing replenishment of the medium and short chain fatty acid substrate. With that being said, I hope I've convinced you that this compound ivacadine is capable of inhibiting long chain fatty acid oxidation, which indirectly hinders mitochondrial respiration as well. We'll be continuing to focus our efforts on finding the molecular target of this compound using some of the experiments I outlined here, while focusing on the 4 that our collaborators' experiments highlighted to us. Lastly, I'd like to thank my own lab group and our collaborators. Thanks.
Questions? All right, thank you. So our final abstract speaker for this afternoon is Adrienne Bianco, and she is from the Department of Clinical Studies.
Okay, thank you very much. My name is Adriana. Like Michelle announced, I'm from Clinical Studies. Today I'll be presenting some results from an owner-reported survey on diet and nutrition in healthy dogs and dogs with cancer that I've been working on. So first of all, a little bit of background information. Studies have shown that most dogs seem to consume a commercial pet food, so about 90%, for half of their diet or more. And dry food was the most popular diet type in this study. And then there were about 50% of dogs that were reported to be fed only dry food. In contrast, about 17% of dogs consumed a non-commercial food for about a quarter of their diet or more. This included things like homemade raw and homemade cooked foods. And then about 3% were eating exclusively homemade food. In cancer, the picture seems to be pretty similar. We see most dogs eating a commercial food, in this case either dry or wet, and then a smaller percentage getting only homemade raw or only homemade cooked, and then slightly larger percentage receiving a combination of commercial and either homemade cooked or homemade raw. Now, when we switch gears and look over at nutritional supplements, we see about 9% of healthy dogs receiving a nutritional supplement. And then this percentage increases to about 13% when dogs have some sort of disease. However, in cancer, we see a bit of an increase here, with one study citing that 40% of dogs with cancer received a nutritional supplement, and another confirming this with 35% of dogs receiving nutritional supplements. Then 48% of these were found to feed supplements because of the dog's cancer diagnosis. So this brings me to the objectives of my study, to compare supplement use and diet type in dogs with cancer and healthy dogs, and compare sources of information consulted by owners of dogs with cancer and healthy dogs when researching diet and nutrition topics. For hypothesis, I'm expecting number of supplements fed will be higher among dogs with cancer compared to healthy dogs. Dogs with cancer will be less likely to be fed a conventional diet, and owners of dogs with cancer will favor different sources of information compared to owners of healthy dogs. So, the study design, we started with a power calculation to determine our minimum sample size. That was determined to be 102, so 51 in each group, for a significance of 0.05. We designed the survey using Qualtrics and used 52 multiple choice and fill in the blank questions. And then we received our Research Ethics Board approval and began recruitment. For recruitment, we started recruiting dogs for the cancer group in the Moana Campbell Center for Animal Cancer. This ran from October 2018 to February 2019. This was in-person recruitment. For the healthy group, we were recruiting at the OVC Hills Primary Healthcare Center, again, October to February. And then to boost recruitment a little bit, in January to February, we did also use social media to recruit. For statistical analysis, we used SPSS, and frequencies were reported for demographic data. Chi-squares and odds ratios were used to analyze categorical data, and then Mann-Whitney U-test for continuous non-parametric data. So this is what my sample ended up looking like. We had a total sample size of 389, so we did exceed that minimum sample size. Owner demographics and dog demographics were similar to other studies that were done in this similar field, so that was good to see. For diet data, first of all, I'll just explain the color scheme here. So from now on, data representing healthy dogs will be in blue and dogs with cancer will be in orange. So we did find that in the frequency data, it seemed like dogs with cancer favored feeding homemade cooked foods and homemade raw foods compared to healthy dogs, whereas the healthy dogs seemed to more commonly feed the conventional dry food compared to dogs with cancer. So we ran the stats on this and found that this was a significant difference. Dogs that were healthy were about 4 times more likely to receive commercial dry food, whereas dogs with cancer were 2 times more likely to be fed a raw food and about 4 times more likely to receive a homemade cooked food compared to owners or compared to healthy dogs. Looking at previous studies done, this is a little bit different. In 2016, we see that the number of dogs fed, or percentage of dogs fed dry food, seems to have decreased, and owners seem to be favoring homemade cooked foods and homemade raw foods currently. In terms of sources of information, this is the dogs with cancer. We asked where they look for information when researching nutritional supplements, pet nutrition, and pet health topics. 2 interesting things here. We see the veterinarian is the #1 source of information, so that's good news. However, social media groups was very close second in most of the things we asked about. So again, looking at the stats, we did find that owners of dogs with cancer were about 4 times more likely to consult social media for information about pet health and nutritional supplements, and about 3 times more likely to consult social media for information about pet nutrition compared to healthy dogs. And then looking at the study from 2006, we see the veterinarian is consistently the most popular source of information, so that is, again, good news. Friends and family seems to have increased a bit in my study, and then the internet is definitely something that's starting to increase compared to 2006. Looking at the healthy dogs now, we see some similarities and some differences. Veterinarian is still #1 source of information, so that's great. Social media groups is seemingly decreased in this group, and then we do see pet store seems to be slightly increased. So looking at the statistics, we did find a significant difference here, where odds of consulting pet stores for information about pet health and was about 3 times higher, or 3 times more likely for healthy dogs compared to owners of dogs with cancer. And then about 2 times more likely to consult pet stores for information about pet nutrition and nutritional supplements. So, seems like owners of dogs with cancer and owners of healthy dogs are consulting different information for nutritional supplements in particular. So, next I looked at nutritional supplement differences between these groups. So, in the group of dogs that do not have cancer, of 257 respondents, 110 reported feeding nutritional supplements. So that's 43%. In the group of dogs that do have cancer, of 132, we saw 86 report feeding nutritional supplements, so about 65%. This difference was found to be significant. Owners of dogs with cancer were significantly more likely to feed nutritional supplements. Then when we look at the range data, this is the number of supplements fed per dog. So in both groups, the minimum number of supplements fed was 1, which makes sense. And then the maximum was higher in the dogs with cancer. So we see a range of 14 versus a range of 19. And the mean number of supplements fed was also increased in the dogs with cancer. So this difference was also found to be significant. Dogs with cancer were receiving more nutritional supplements than dogs that do not have cancer. So this next led me to look at the types of supplements being fed. This is a graph of the total reported supplements, all grouped into similar categories between dogs with cancer and healthy dogs. So there are definitely some trends among the 2 groups here. When I ran the statistics on this, I decided to look at some of the most popular supplements fed between the 2 groups and see if there were any significant differences. So over here, we have the supplements that were found to be significant. CBD oil, turmeric and curcumin, mushroom supplements, and supplements containing a multitude of different compounds that was marketed as an immune-boosting supplement were all more likely to be fed to dogs with cancer compared to healthy dogs. Supplements like fish oil, omega-3s, multivitamins, multi-minerals, probiotics, and glucosamine or chondroitin were all popular among both groups, but there weren't any significant differences between healthy dogs and dogs with cancer there. In terms of previous studies, here we have the study from 2016 again. These are a few of the supplements that were similar in both studies, and there are some differences and some similarities, but I think most prominent here is herbal supplements seem to be on the rise compared to that study. So before I wrap up, I'll just revisit my hypotheses. Number of supplements fed were higher among dogs with cancer compared to healthy dogs. Though the frequency data did indicate that dogs with cancer were more commonly being fed dry food, we also did find that dogs with cancer were more likely to be fed a homemade raw or a homemade cooked diet when compared to healthy dogs. And then owners of dogs with cancer and owners of healthy dogs were both using the veterinarian as their primary source of information, but we did find that social media was favored in the healthy dog, or in the cancer dogs compared to the dogs with cancer. So for future directions, definitely more nutritional research is needed in these supplements, especially research pertaining directly to dogs. We don't really know a lot about the safety and the efficacy of some of these supplements. And definitely client and clinician communication and education when it comes to consulting on online sources and making sure that they're using reputable sources of information when making their dietary decisions. There's a couple acknowledgements here of the people who made this study possible, some of the references that I used throughout this presentation, and I'll take any questions now. Yes.
I was wondering if you reported the ages of the dogs in the study. So the dogs with cancer be a much higher age group, which might explain some of the different age regimen?
Yeah, so I did do that. I think it's in one of my supplemental slides here. Bear with me. All right, so here are the ages. This is also compared to that 2016 study. So dogs with cancer were slightly older than the healthy dogs, so definitely could be a conflict in some of the supplements that we saw fed. Yep.
So do you think these results may reflect the type of owners that we welcome around? It's like the demographics, the way people look at things, the way people get information.
Yeah.
Compared to just looking at this small population in Ontario to, let's say, an international study that has been done on this.
Yeah, great question. In the end, we weren't actually very successful in recruiting at the cancer center, so most of the dogs in the survey were from different areas. I have the specifics on that a little further back here. Oh, maybe not. So we didn't get too many from Guelph. Most of the respondents were actually from the United States, and then Canada was the second. So seems like it's a bit of a more broad picture there.
Any other questions? Great, thank you.
Okay.
I'm very excited to announce our keynote speaker who's representing his busy schedule in Vienna, China, Wisconsin, and now Guelph. And so David Vale is originally from Edmonton.
Yay. Thank you, thank you.
I'm a graduate of the Western Veterinary College at the University of Saskatchewan.
Woohoo!
Vic did his postgraduate training at the University of Wisconsin-Madison. Badgers. And board certified. He's now the professor in the Suran Comparative Oncology position chair at the University of Wisconsin-Madison. He's also a member of the Carbone Comprehensive Cancer Center at the University of Wisconsin as well. When Dave first went there, he was working with with Greg. And so Greg McEwen, who is a pioneer in the world of veterinary clinical trials, Dave's done a great job at taking that and going much further with that. And we're going to hear today about some of the clinical trials, but as you can look through his bio there, he's got numerous publications. He's been awarded research awards such as the Mark Morris and the Pfizer Research Awards. He also has been on many advisory committees. He's also done a great job at the leadership for the veterinary group. He's been past president of the Veterinary Cancer Society, the genomics.
And yet he's so young looking.
And yet that's why and yet he's so young looking.
Thank you. Appreciate that.
So David today, though, is also going to talk to us about clinical trials, cancer immunotherapy trials. And I'd also point out, though, he's also the co-editor for the textbook Clinical Small Animal Clinical Oncology, which will be coming out in
October.
In October, shameless plug.
6th edition.
And when he's not doing all that, he's also
royalties.
a grandfather to 5 kids.
5 grandsons, a hockey team.
So without further ado, let me introduce you to Grandpa Vale with your talk.
Thank you. Appreciate that ever so much. Thank you. Well, I really appreciate the opportunity to come and speak in sunny Guelph. It's always a great opportunity to come back to Canada for a couple days, wash the Trump off, and then head back down there. Sorry. Hopefully there's no ICE agents in the crowd. So yeah, we're going to talk about kind of a one medicine approach to cancer immunology, and obviously cancer immunotherapy is a huge topic, and I'm only going to be speaking on a few portions of it, but I'm going to kind of discuss the overriding problem, what the potential opportunities are for a comparative approach, what the obstacles are for a comparative approach. Just a couple of slides on that because this group is very well versed in that. And then kind of lead you through some of the trials that we're currently running with a one medicine approach with our cancer center with new immunotherapeutics and diagnostics that will help predict response to immunotherapeutics as well. So if this works, which it may, I actually pulled this out of the Ottawa Business Journal. This is my Canadian content other than me for this particular talk. We all know how important immunotherapy is becoming, and over the last few years, immunotherapeutics have been voted the advancement of the year at ASCO the last 3 years. So big, huge topic, a lot of potential in this area. So the problem, kind of the overlying problem, and it's been talked about at several points through the talks throughout the day, is that for the vast majority of cancer patients throughout history, a diagnosis of metastatic disease is essentially a uniformly fatal condition. And that's where we really need the help. Dealing with primary tumors, we can apply surgical techniques, we can apply radiotherapy techniques. It's the systemic disease that ultimately gets me, or gets people with cancer. But in the last few years, we've recognized a minority, a subset of individuals where immunotherapy is creating spectacular and durable responses. And really, the problem ahead of us is trying to change that minority into the majority of patients. So could the comparative approach accelerate this process of moving a minority population that's responsive durably to the majority? And again, I'm not going to spend much time on this. You've probably all seen this particular publication where many of the potential opportunities by including companion species with cancer in our cancer development, drug development, but I've highlighted the ones that are particularly important from a standpoint of immunotherapy. Certainly intratumoral and inter-individual heterogeneity cannot be recapitulated in most models for the most part. We really need a larger species that's developing naturally occurring spontaneous tumors. Certainly the complex interaction between tumor and the immune system and the tumor microenvironment, so important to what we're seeing and what we still are really quite vague about, and where we need a lot of improvement in our understanding of the biology that's involved. Certainly a syngeneic microenvironment as well. I also threw up and added to this slide body size. It allows us to use the same types of radiation therapy machines, the same types of advanced imaging modalities, and I'll be speaking a little bit about molecularly targeted radionuclide therapy today as an immunotherapeutic. We really can't do that kind of work in mouse models just spatially, depending on the wavelength of the particular nuclide particle. So that's really important to us as well. So what are the obstacles to including companion species? When we all got together in DC in 2015 at the Institute of Medicine for these, it was a big get-together of physicians, veterinarians, journalists, lay people, basic scientists. How can we advance this one medicine approach? One of the aspects that came out of that meeting was that our toolbox is really limited, especially for immunotherapeutics. We don't have the right antibodies. We don't have antibodies that work well with flow. They might work well with immunohistochemistry. We've got a lot of work to do there. And our infrastructure, our sharing infrastructure, there's several silos across the North America that are working in little individual focused areas that just aren't communicating. So out of that came a number of helpful infrastructure changes, and these are some of them. This cancer moonshot, big thing in the United States, where it was recognized that we really need to do 10 years of work in 5 years or 20 years of work in 10 years, just as the Americans set out to get to the moon in a short period of time. And out of that came some tremendous funding opportunities. The U01 infrastructure, which funds multi-center data sharing, very large grant opportunities for research that include companion species. One in particular is strictly based on canine immunotherapy, and there's a central agency set at the University of Pennsylvania and several other major academic centers that are involved in projects within that infrastructure. And this is all data shared. We have a U01 that is involved in radioimmunotherapy, and that's what I'm going to spend a fair bit of time talking about today. But we all get together monthly for teleconferences. We share information. As somebody discovers a new monoclonal that works good for the dog in this scenario, that's all set out for us so that we can really, short of intellectual property, get access to that new information. Certainly there are other consortia around, the CORC, the Comparative Oncology Research Consortium, which involves 5 centers. There are academic centers that also have a National Cancer Institute recognized comprehensive cancer center associated with them and a veterinary school. And in order to apply for those grants, which the fiduciary body for that is the V Foundation. I don't know if you know what the V Foundation is. It's a huge private foundation started by ESPN that will fund these trials. There has to be a PI from the veterinary school and a PI from the Comprehensive Cancer Center in order to apply for these grants. And then some of us were in this room, we're at the Kids and Canines meeting. That's another 503c foundation that funds this type of research as well. So we're getting there. Our infrastructure is much better, and we're kind of coming out of our silos and getting together. So a lot of what I'm going to talk about is this whole concept of immunosurveillance, and that's what we're really trying to maintain and re-energize in ourselves and our patients. And that's certainly the ability of the immune system to identify abnormal cells, cancer cells, and clear them before they develop into full-blown cancers. And the advantages are pretty intuitive, that you can target cells anywhere in the body, and importantly, you can create memory such that you can deal with metastases or recurrence at a later date. So most of you are familiar with Robert Schreiber's lab, and many others came out with this concept of the dynamic equilibrium between cancer and the immune system. And initially, this is what, of course, we want, is we want elimination. So the immune system is out surveilling, sees a cancer cell and kills it. But eventually, through processes like immune editing and several others, we reach an equilibrium where the cancer is kind of in a precancerous form living amongst us but being kept in check. But eventually, in some cases, we get escape ultimately, and the cancer takes off, the immune system is suppressed, and we develop full-blown cancer. And there's points of translational opportunity all along here where we could intervene. I'm going to spend most of the time today talking about immunotherapy, but I'll spend a little bit of time at the end, if there's time, talking about some of the new preventative vaccine technologies that are coming down the pipeline. And unfortunately, this idea of immunosurveillance, as we age, this process of immunosenescence kicks in, where our immune system essentially gets old and tired like we do, like grandpas, like me do. And not only are we more prone to developing infections, but we have reduced capabilities for immunosurveillance, and eventually we have escape from our cancer cells and ultimately develop full-blown cancers. So we want to try and reverse this process and reverse the immunosenescence as well if we can. So those are the problems, the opportunities and obstacles, and how we're trying to overcome those with infrastructure. I'm going to kind of tease out some things that we have done over the years at University of Wisconsin, which, by the way, University of Wisconsin serves as the farm team for your Canadian Women's Olympic team. I don't know if you've noticed that 5 of your players are our Badgers. We just won the national championship. The captain of the team is going to be a veterinary student starting next year, so that's awesome for us. Our Friday night student-faculty games are going to become quite interesting. And as Paul mentioned, where did Paul go? Look for the Coke bottle. So I came to University of Wisconsin because of Greg McEwen, who really was the grandfather of canine immunotherapy trials, comparative immunotherapy trials. Many of you are familiar with Muramil tripeptide trials for canine osteosarcoma that moved into children. And he was really instrumental. He was the one that told us, it started telling us as clinicians that we should really be getting behind clinical trials. And this is my campus, University of Wisconsin, one of the prettiest campuses, I think, in North America. It's on an isthmus between two huge lakes. It's kind of a party school for the undergraduates, but the rest of us get a lot of work done, I think. Pretty sure. Okay, so as far as our investigations, in a more global sense, what constitutes a clinical response to immunotherapy? And that's always been a problem. And when you look at some of the new drugs that are coming down the pipeline, the checkpoint inhibitors, they're very expensive. They're not devoid of side effects. But because of that, we'd like to be able to know whether a person is responding quicker, sooner rather than later. Again, because of cost, and we want them to be on an effective drug. Well, one of the problems, and some of you are familiar with the work that Wilczek published, he's also the doc at Sloan Kettering that was involved with the canine melanoma vaccine development with Phil Bergman. But in human trials looking at checkpoint inhibitors and anti-cancer vaccines for melanoma, they realized that the way people respond to immunotherapy is different than the way we respond to cytotoxic chemotherapy. So in the upper left panel, that's kind of the standard way that people respond to cytotoxics. You give a series of drugs and the tumor shrinks down, so the y-axis is the size of the tumor and the x-axis is the period of time. And so you give drug and the tumor shrinks. That's kind of the standard cytotoxic approach. However, people that are on immunotherapy can go through, a significant percentage can go through what's called pseudoprogression. That is, that their tumors get bigger before they get smaller. And a lot of those patients are being thrown out of trial when they're actually on an effective drug because they're deemed to be failing if we use the old RECIST, the response criteria for solid tumors based on size. Also, with immunotherapy, the responses tend to be delayed, right? It takes a while for we turn on the innate immune system, and then the adaptive system takes some time before we get a true specific immune response. So it's much more delayed than what we are typically see with our cytotoxics. And then the other thing is that with immunotherapy, you can develop new lesions before you start to see response, and those patients would also fail based on the old RECIST criteria. In this patients that are on immunotherapy that develop new nodules, they pull them out and histologically assess them. There's actually, the tumor is not getting bigger because the tumor is proliferating. The tumor is actually necrotic, and it's all inflammatory component effector cells of the immune system. So we need to be able to recognize that early so we know who's responding, who's having pseudo-progression, and who's failing the drug and move them on to something else. And if you look at, well, I'll just, I think I've made the point, so I won't go into that. So one of the things that we've been working with our medical imaging and medical physics group is a way of using advanced imaging to determine early, and I'm talking within a week, who is responding to an immunotherapeutic and who isn't. And this brings us to the topic of radiomics, which is the use of relatively standard, it's certainly standard in physician-based oncology and a little bit exotic in veterinary-based oncology, CT, MRI, PET-CT. And taking textures, creating hundreds of thousands of pieces of data from that imaging, and then creating algorithms with bioinformatics to determine certain or answer certain questions about that particular patient. Now, just a really simplistic model is something that we've done at University of Wisconsin, where we took 172 CT images. So this is standard CT imaging. Created an algorithm based on these textures that we can pull out of the images. And we can actually predict who's going to survive and who's going to be a long-term survivor and who's going to be an early failure. But we can also predict what the histology is. Apologies to Jeff, but we can get rid of the pathologist into the equation. We can tell who has a squame and who has an adenocarcinoma based on radiomics. It's very advanced. If you add functional imaging to that, like PET-CT, on the human side, they now have algorithms where they can scan a person with lung cancer and determine, based on their texture, their standard scanning, whether they're EGFR positive, whether they will respond to a particular small molecule inhibitor without any pathology, without any immunohistochemistry. So if we could do this, it's certainly non-invasive, right? We don't have to go after biopsies, and we can personalize medicine based on an image. Wouldn't that be fascinating, or wouldn't that be spectacular, right? So Robert Uri, who's a medical physicist at University of Wisconsin, we've worked together for years on other projects, wanted to come up with this immune response radiomics. So let's apply the field of radiomics to immunotherapy and see if we can pick up some textures, some techniques that could tell us who very early is responding and who isn't. And the problem is that we're trying to discriminate between inflammation, which will increase glucose utilization. So the standard cancer radionuclide that's used in PET-CT is FDG, a glucose nucleide. Using a second radionuclide, FLT, which is a radioactive thymidine, is only picked up by proliferating tissues. So if you have a lot of inflammation and a lot of proliferation, you should see actually both FDG and FLT go up. If you're actually killing the tumor but increasing the inflammation, only your FDG should go up, right? So we use a ratio to try and be able to discriminate between pseudo-progression and actual progression. That's essentially the idea behind it. So our first patient, our index species case, was Fletcher. Fletcher had actually been involved in 2 clinical trials prior to this one. You can tell that he's amputated, and he developed a stump recurrence. So he was treated in the 555 Morris NCI trial, looking at rapalogs for amputation, carboplatin, and rapalog versus control. And he went about 16 months and recurred. He had a stump recurrence, which is very uncommon in veterinary oncology. But he developed a stump recurrence, so we said, well, let's see what we can do. We know we're going to go after this surgically, so we'll get access to all these tissues afterwards. So we took them to our PET-CT, PET-MR units, and we created an FDG-FLT ratio with tremendous numbers of textures, literally millions of data points based on, we have all the textures from CT, we have all the textures from MR, and we did MR FLT and CT FDG. So we have the FLT and we have the FDG textures as well. And we create essentially histograms that we can go back and correlate with the tumor once it comes off to validate our imaging. And what you essentially do, this is Fletcher in the scanner scanning back through his stump recurrence. On the left is the FDG scan, changes over time. That's before treatment and a week after treatment. In the middle is the FLT, so that's the thymidine. Those are proliferative tissues. And I don't know if you can see this, but the left panel, which is FDG, over time, as you go through the tumor, it's becoming hotter with respect to FDG. So there's something that's very metabolic there. But in general, in the middle panel, which is FLT, it's becoming bluer, which is colder. And that means that there's less proliferation overall. And then you create an FDG-FLT ratio. And you essentially come up with a heat map based on all these textures, just like looking at a genome heat map or an epigenomic heat map. And based on this, we come up with an algorithm to tell us, is this inflammation or is it pseudoprogression? Is it proliferation of tumors? And again, we have the advantage that we can validate with the entire tumor. So we can certainly do that with any tumors that we're going to subsequently remove. Otherwise, you have to take serial biopsies. And we can correlate this as we did with changes in infiltrating lymphocyte populations, circulating lymphocyte populations, gene expression of those lymphocyte populations as well. So hopefully, some of these algorithms will get so sophisticated that we'll be able to tell if they're having an immune response and in what way are they having an immune response. Case number 2 of this immunoradiomics, this is Dino. Dino had a metastatic melanoma into the pre-scap lymph node. We scanned him before and after, 7 days after intratumoral immunocytokine, and I'll talk about that particular trial in a second. But just suffice it to say that we're trying to stimulate an immune response in that tumor. And if you look at his baseline on the left and his 1 week post-treatment on the right, FDG on the top and FLT on the bottom, you can see that this particular dog, Dino, is having a phenomenal anti-proliferative response within a week as picked up by functional PET-CT imaging. And so that's great. We know that we're causing anti-proliferative effect in the tumor, but we're having a proliferative and a metabolic effect in his regional lymph node. So Dino is the stereotypical, this is a positive immune response. So obviously, this is early, and all of our subsequent trials, which I'm going to talk about in a second, are going to be using this radiomics approach for a validation of the process. Okay, so that's problem number 1. Problem number 2 is, of course, that tumors are really, really smart and are really, really hard to treat because they start to escape the immune system. And the mechanisms that they do that, I don't have to tell this group, that there are multiple. The ones that are getting all the press right now, of course, are the checkpoint inhibitors. And at least in the US, I can't watch the national news. Well, I can't watch the national news anymore anyway. But you can't watch the national news without seeing direct-to-consumer marketing of the checkpoint inhibitors, the Keytrudas, et cetera, anti-PD-1 monoclonals, anti-CTLA-4 monoclonals. And we have those. There's a canonized anti-PD-1 coming down the pipeline that we were involved in a national trial that finished last year that's undergoing regulatory approval right now. So that'll be awesome for us, and we can start applying that to our immunotherapy trials as well. But we really need to worry about this tumor escape from the immune system. So we can create all the effector T cells we want that are all very specific for the tumor. They get into the tumor microenvironment and they get shut down. The tumor is really good at, by itself and also by educating the tumor microenvironment to put the brakes on to the immune system. So ways to get around that. I'm going to use primarily examples of a combination of radiation therapy and immunotherapy to illustrate how we as an institution are pushing the envelope on this one medicine approach. Many of you know Lisa Forrest. She's headed up our radiation therapy department for quite some time, our tomotherapy unit. And what we're after, obviously, in the end, what everybody's after is this curative potential, the interface between radiation therapy and immunotherapy, can we bring those two modalities together to get enhanced, durable responses? And the two have really worked in separate silos for the last century. But they've been occurring in parallel in similar ways. That is that radiation therapy and immunotherapy started at the turn of the century. They were applied in the clinical setting starting in the 50s really and moving forward, but in a very unfocused way. For example, as far as immunotherapy is concerned, it was all just non-specifically stimulating the innate immune system. But now we're getting into more specific adaptive immune system technologies as far as our immunotherapy. And the same holds true on the side of radiation therapy. Much more specific, conformally avoiding types of radiation therapy that are hopefully used together be quite beneficial. So I'm going to talk a fair bit about this in situ vaccination effect. That is that it has been shown, and we are showing that now in our clinical trials, that we can turn a patient's own tumor into a vaccine against the cancer by manipulating with various immunotherapeutics like checkpoint inhibitors, cytokines, monoclonal antibodies, and radiation therapy to create that environment where we will actually create a therapeutic vaccine situation that will hopefully help the majority of patients rather than the minority. Not that we want to get rid of the surgeons. We'll still need you eventually, right, to clean up what's left behind, the necrotic husk of what's left behind after we're done. All right, so how does radiation therapy overcome the mechanisms of tumor escape? By several ways. And it's a timed event. That is that if you look on the left here, I put seconds, hours, days, and weeks as post the radiation therapy event. Initially, we have things that happen very quickly, like depletion of lymphocytes within the radiated field. And that's both types of the good guys and the bad guys. So the Tregs, the suppressor cells, the myeloid-derived suppressor cells, etc. And we also get rid of the effectors, the CD8s, the ones that we want to be there, right? But very quickly after that, the tumor cells themselves and the tumor microenvironment start to change the release and the type of cytokines that are released that will eventually bring back the effectors. And that happens very quickly. That happens within hours and days. And we also alter the tumor itself to express new antigens, to express more levels of MHC, for example. So there are various mechanisms, much more complicated than we have time for today, where we change the phenotype of the tumor cell to become more immunogenic. And then the tumor cells, of course, die, and we create DAMPs, or death-associated molecular patterns, which are also recognized by the immune system and get fired up. And ultimately, we hope that we create dendritic cell maturation, antigen cross-presentation, and this is important and it has been documented, diversification of the T cell response. So if there were, say, neoantigens within that tumor or tumor-associated antigens within that tumor, and there wasn't a T cell subset targeting that after radiation therapy, there may be, and I'll show you some data for that. But we really don't know much about this process. There's how much radiation do we give? What type of fractionation protocol do we give? If we're going to be combining it with immunotherapy, when do we give the immunotherapy? When do we give the radiation therapy? Those are all questions that are, for the most part, unanswered, where we, as veterinarians, can include our companion species in these clinical trials to help answer some of those questions. So at the end of the day, you've got the unirradiated half of the tumor here and the radiated half, and there's tremendous differences that are well characterized. Depletion of the T regulatory cells, upregulation of FAS, MHC, chemokines, all things that should result in a more robust immune response to the particular tumor. And so this idea of an in situ vaccine is really a personalized approach to treating a particular patient. How can you get more personalized than using the patient's own tumor to create a vaccine, right? I mean, that should be the ultimate way to go. So I'll show you some data first in mice, and then we'll roll into dogs, and we'll go from there. So this whole process of radiation helping the immune system came about by when it was recognized this idea of the abscopal effect. And the abscopal effect, as shown in this particular dog, you have a dog with a primary tumor who unfortunately has metastatic disease. If you irradiate the primary tumor, which is what we often do, and say these are They could be micrometastatic disease or macro disease. This is the typical response. 99% of the patient population will have a nice response at the primary that you include in the radiation field. And unfortunately, the metastatic disease continues to grow away merrily. But in less than 1% of patient populations, we notice that if we irradiate the primary site, the metastatic tumors get smaller as well. And that's called the abscopal effect. It's a very rare event, but it's well documented over the years. But you can see how rare, looking over almost a 50-year period, there are 46 publications on the abscopal effect. So how can we improve on that? How can we make this 1% of patients 100% of patients? That's through various techniques, one of which is just adding radiation therapy to the primary and then starting to manipulate what happens afterwards. But again, we need a lot of questions answered as far as dosing, when we apply, what's the sequence of applications of our immunotherapeutics and our radiation, and what doses. So if we look at some things that are associated with how we deliver radiation therapy, it's been shown that if you used our standard hyperfractionated approaches to radiation, so a protocol that lasts 4 to 7 weeks where you give a small dose of radiation daily, allow the normal tissues, of course, to heal, and you keep doing that over a long period of time, that's lymphodepleting. That's been shown that as especially with large volume tumors, as the lymphocytes circulate through the system and they get irradiated every day, we drop down in numbers of lymphocytes. If you give one dose or a hypofractionated protocol, maybe one dose a week for 4 weeks instead, you don't see lymphodepletion. And lymphodepletion is definitely, and this is just one example, but definitely been associated with poorer outcomes. So this would imply to us that using hypofractionated approaches for immunotherapeutic synergy might be more appropriate. Another example of applying different dosages, and so that was kind of a frequency, a little brief glimpse of frequency, would be dose. We know when we irradiate tumors or cell lines, whatever, with radiation therapy, we create micronuclei, and that's actually what's been used since the 80s for dosimetry. You count the number of micronuclei, and so this is a documentation of what dose you're delivering. But this is dose responsive. The micronuclei go on to produce double-stranded DNA in the cytosol, and it shouldn't be there, right? Unless you have a virus, a viral infection, and that stimulates this viral response pathway, illustrated here on the right, usually through the STING pathway, but there are others where we change, if you have double-stranded DNA in the cytosol, STING is upregulated, and you get an interferon type 1 approach. So interferon gamma, for example, which makes the tumor cells more sensitive to the immune system, stimulates the immune system. But it's dose dependent. If you get above 12 gray as a single fraction, you start to drop off on the level of cytoplasmic double-stranded DNA, and that's due to the T-Rex exonuclease system. It will kick in and start chopping these up, and then we lose, it abrogates our effect, and so we get less of a viral response pathway. So again, that's another example of how do we deliver? How much radiation is right? Do we reach a critical point? And those are all questions that we can take from the lab and bridge that data in our companion species to answer some questions. I've already talked about T-cell repertoire gets increased, so I'm not going to spend any time on that per se. The other, and this is kind of the classic thing that has come out, and this is where radiation has a lot of tremendous potential, is taking immunologically cold tumors and turning them into immunologically hot tumors. That is, that by irradiating them, we can make them responsive to immune therapy. Classic example is some of the earlier checkpoint inhibitor trials. In this case, patients that are given chemotherapy are in blue. These are melanoma patients, and patients that are given checkpoint inhibitors in orange. And those with cold tumors all die. And what I define as cold is they have a low mutational burden, and it's been shown that the fewer number of mutations you have, the fewer number of potential neoantigens you have, so the fewer potential for the immune system to recognize something abnormal. They often have low MHC expression and many suppressive immune cells in their tumors when we look at this population. Versus the hot population, these are tumors that have high mutational load, lots of neoantigens around, good MHC expression, and few suppressor cells. And it's been shown, and we're currently working in this area of if you take a cold tumor and irradiate it, in many cases you can make that tumor hot. And this was first actually thought of as an event, this mutational burden issue, because the people with lung cancer that respond the best to the checkpoint inhibitors are smokers. And that's because they have huge mutational burden in their tumors because of years of smoking. So that was one of the things that first led them to look at this particular phenomenon. Okay, so we're going to use radiation therapy in our clinical trials, and we're going to consider adding, because nothing alone is going to, and we already know if all we do is radiotherapy, we're only helping a small subset of particular patients. So other avenues we have are certainly monoclonal antibodies, and again, I probably don't have to tell this group how monoclonal antibodies work, primarily through ADCC and complement-dependent cytotoxicity as well, where we get engagement. You can have cytotoxic T cells, NK cells coming in and essentially destroying the tumor cells. So we can do better than that, though. We do have some monoclonal antibodies, certainly on the human side, and we're getting more caninized monoclonal antibodies on the canine side that will be more effective and repeatably usable. So we've been working actually for several years with a group at University of Wisconsin with an immunocytokine, which is a fusion protein that fuses a particular monoclonal antibody that recognizes a tumor-associated antigen with interleukin-2. It cuts down the systemic toxicity of interleukin-2 because it specifically targets it to whatever your target is. And in this case, we're using a target which is GD2, a ganglioside that is often expressed in melanoma, both in humans and in dogs, soft tissue sarcomas, and osteosarcoma. And so in this case, not only do you get what you would traditionally expect to see from a monoclonal antibody, but you also get the enhanced IL-2 effect that's local in the tumor microenvironment. So a group that we've been working with, Zach Morris at the Carbone, he's a radiation oncologist, looking at if you take a mouse and you inject it with a GD2 positive tumor, in this case B78 melanoma, and you irradiate it, and then plus or minus give that immunocytokine intratumorally. So you're giving this immunocytokine into the tumor. You can give it systemically as well, but for some of the problems that were brought up by Jim's group, some of these intratumoral pressure issues, for example, intratumoral actually, we get some really nice responses. And if you look at patients, these are rodents, that are given intratumoral immunoglobulin alone, not the fusion protein, but just anti-GD2, versus those that get radiation alone, versus those that get the immunocytokine alone, versus those that get the combination. We're getting about 70% disease-free. Now, of course, it's a rodent, and you give me a rodent with a tumor, I can cure it with just about anything, right? Interestingly, if you wait 3 months and you take this cured population and then you go ahead and challenge them with their tumor, there's memory there. They don't engraft with the tumor. Whereas if you compare it to mice that you just excise the tumor surgically, and then you challenge them 3 months later, they of course all engraft. So we have an immune response to the tumor, and we have memory created. So that's great. And so again, kind of summing that aspect up of using the immunocytokine, which has several positive immunotherapeutic benefits, ADCC, calling in NK cells and effector T cells, get some antigen cross-presentation, and engagement with the IL-2 and activation of those effector cells that you've called in, and then all the effects that we've talked about with radiation therapy, just briefly of course, but changing MHC expression, the way antigens are presented, upregulating FAS, depleting T regulatory cells, etc. So in combination, those are all potential areas why we're seeing such good responses. So let's bridge that. We've got the rodent work, let's bridge it to our canine work. We applied for a Veterans Administration grant because veterans who serve in the Middle East have a higher melanoma rate than the general population in the United States, primarily because of sunlight exposure. And we were working with Paul Sondel, who's a pediatric oncologist, immunologist at UW, and Mark Albertini, who runs the physician-based melanoma clinic there. And we applied for funding to start looking at this process in our dog population. The first thing, of course, we had to document for our grant was that indeed the GD2 is expressed in the tissues that were in the tumor types that we're going to look at. You can see melanoma, this particular osteo is quite weak, but soft tissue sarcomas, a lot of GD2 to target. And so this is what we're doing, although the mix is being switched. We're using different fractionation protocols, different dosages. And so we start with, of course, our biopsies and these are all biospecimen heavy trials. Go ahead and deliver coarse fraction or hypofractionated radiation therapy. Once we have the anti-PD-1, we have an arm that will be added to this mix. And then about a week later, they get their intratumoral immunocytokine. And this is based on, the timing is based on rodent work, but we will be altering that in various arms of the trial. And then follow them with biospecimen-rich follow-up at that point. So this is, again, and all these dogs are undergoing the radiomic analysis as well, so that we can hopefully distinguish who's responding early rather than late. So that's one example. How can we better apply these technologies? The interesting thing is that, yeah, those dogs have a primary tumor, and Michelle's going to argue to me, just cut the primary tumor out, right? Don't need to worry about all that other stuff unless it's greater than 2 centimeters or whatever the prognostic factor that you're using. But that's not the scenario that we worry about. That's not our primary. If you remember the very first slide or second slide, our primary problem is people and dogs with metastatic disease. So what if they have a second tumor? How does this kind of affect the system? So Zach's group has actually done a lot of rodent work with this, which is really quite interesting. You take a mouse and you give it the B78, GD2 positive melanoma. And then you inject on the opposite flank either more B78 to get 2 tumors or a different tumor type, pancreatic. And the pancreatic that you use can be GD2 positive. So you've got the same target, but a different tumor, okay? Then you block them and you irradiate the primary site. You give the intratumoral immunocytokine just to the primary, and then you follow them. And interestingly enough, yeah, we saw what we saw in the earlier study. If they have the B78 primary alone, we get cures. If you have the B78 primary and an irrelevant tumor, the pancreatic, even though it has the target on it, you still get the effect of the primary tumor. But if you give them the same tumor in another site, i.e. a metastatic lesion, you lose your effect. Okay, so you don't see anything abscopal. And it's the same if you use the different tumor. If you use a GD2 positive pancreatic and you give them on the opposite flank, either pancreatic or the GD2 positive melanoma, if they have the pancreatic on the other flank, they still die of their tumor. So what's going on there? And this has been called concomitant immune tolerance. That is, that there's something about that metastatic lesion that's causing your in situ tumor effect in your primary to fail as well as in your metastatic disease. There are a couple of theories behind that, and some have been backed up with data in that the metastatic lesion is repopulating the primary with the T regulatory cells that you've depleted and some of the other immunosuppressive cells that you've depleted. Or the effector cells that you create are circulating and being re-educated in the distant sites, and the brakes being put on by the cancer cell and the tumor microenvironment in the distant sites. And if you look at that, we have data to suggest that that indeed is what's going on. If you look at the ratio of CD8 positive FOXP3 positive cells, so the FOXP3 are Tregs, are immunomodulatory cells, and the CD8s are the effector cells, the good guys. If you have a single tumor and you irradiate it, you get a nice increase in the ratio of CD8 to Treg. But if you have that secondary tumor of the same histology, you get an initial spike, but then it's damped within days. So, what if we irradiate the second site? Again, if we irradiate the second site, we reestablish our in situ tumor vaccine, and we get responses throughout the body, including the metastatic. And all you need is a low dose. In this case, 2 gray. So 2 gray is not enough to kill that metastatic tumor. It's just not, unless it was a lymphoma, but it's not. This is a melanoma or a pancreatic tumor. So if you give 2 gray radiation therapy, to the metastatic lesion, we'll call it the metastatic lesion at this point, and then give the intratumoral immunocytokine to the primary, you can reestablish the effect that you had and cure those particular mice. And there's no difference between 2 gray and giving a therapeutic 12 gray to the metastatic disease. So we're reestablishing that in situ vaccine effect in both the primary tumor and the secondary tumor. Just controls without radiation. But the problem here, of course, is you have a patient comes in that has widespread metastatic disease. You can't really irradiate. Well, you could. You could irradiate the whole body, but one, this effect is lost, and two, you have adverse events associated with irradiating the whole body. So how could we irradiate all the metastatic lesions? Well, the way that we're looking at right now is using molecularly targeted radionuclide therapy. And this is something that's been going on for the last decade for various tumor types. If you look at, for example, we do it in our cats with thyroid tumors. We use iodine-131. That's technically molecularly targeted radionuclide therapy. Certainly for neuroblastoma and in kids, iodine-131 with a target that targets neuroblastoma. Radium-223 for prostate, and the Zevalin, which is, everybody's pretty familiar with rituximab, so that's a radionuclide-associated rituximab that targets CD20 lymphoma. Those are all examples of molecularly targeted radionucleotide therapy. And many of these are actually theranostics, that is that we can, once we give them their radionuclide, we can image them, determine where their tumor is, create dosimetry from that image, and then give them more of the agent or a more active agent based on that dosimetry. So a couple of individuals at University of Wisconsin, Jamie Weichert, who has a rollout company from UW Arceus, they've developed a alkyl phosphocholine that specifically targets tumor cells, in that tumor cells have abnormal cell membranes with lipid rafts. It's been shown in over 50 different tumor types, so it's agnostic of tumor histology, that tumors want to build their cell membranes with this particular compound incorporated in it. And we can link this compound with just about any radionuclide that you want. And so for our trials, we're using yttrium, but lutetium could be used. You could even use alpha emitters. But in our particular trials, we use yttrium-86 because it's a positron emitter. We'll give our patients yttrium-86, do a PET-CT scan, have the med physics people come in and do dosimetry, and based on the uptake in all the tumors, then give them yttrium-90, which is the beta emitter for actual therapeutics. So again, we've looked at many, many histologies, bladder cancer, in the dog, osteosarcoma, breast cancer, prostate cancer. We have not found a tumor type that preferentially incorporates the alkyl phosphocholine into it in a rate that will allow us to deliver radiation to the tumors and spare bone marrow. So first, the rodent work. So we take our poor little mouse with 2 tumors. We irradiate one with high-dose therapy, then we give them an injection of the molecularly targeted radiation therapy, and we inject their primary with either our immunocytokine or a checkpoint inhibitor or what have you. And again, the same phenomena occurs. We're delivering about a 2 gray dose to the contralateral tumor and a 12 gray dose to our primary tumor, and we start to cure a significant proportion of those particular mice. Now, the problem with using mice, as you can see spatially here, to be able to tease out tumor microenvironment issues from a radiation standpoint and bone marrow issues, depending on the wavelength of your emitter, you really need a larger species. So we've been funded, well, let me just show you. When you combine those, you essentially get a 100% cure in our rodents. Again, they're rodents. And this is a combination, though, of including the checkpoint inhibitors with the in situ vaccine and the molecularly targeted radiation therapy. Until we have the checkpoint inhibitor available later this year, we're working in this particular area here, where we're hoping to cure around 60% of our patients. And this is the reason they die, is we change it from a metastatic disease to death of the primary as well. That's a completely different issue. I already mentioned that yeah, well, why don't you just irradiate the whole body with 2 gray? Well, we lose that effect when we do that. For some reasons that we understand, but for many reasons that we don't understand. If we can molecularly target the 2 gray specifically to the tumor tissue, that's where we tend to get the really good effect. So this is our index case, Rex. He was the first case to enter this particular trial with the yttrium. And so what goes on is yttrium 86. We take him over to our PET-CT, PET-MR facility. We give him a dose of yttrium 86, and then we image him over time. The med physics people come in and they say, okay, this is how many millicurie you need to give of yttrium 90 to be therapeutic to deliver 2 gray to all the metastatic sites. So this is Rex. This is 2 hours after injection of his yttrium 86. This is 24 hours and this is 48 hours. So immediately after you give the drug, it's given intravenously, you can see that all of the yttrium 86 linked to that NM600, which is the compound that gets integrated into cell membranes, is in the peripheral blood, in the heart, brachial artery, etc. Rex, unfortunately, he had metastatic osteosarcoma to multiple sites subcutaneously and multiple pulmonary nodules. Within 24 hours, however, the drug is leaving the vascular space and being incorporated in all of his metastatic disease. These are all lesions. This was a hind limb lesion, a forelimb lesion, so these are slices at various parts in Rex, and these are his tumors. So this is proof of concept that this particular drug is being incorporated preferentially in Rex's tumors. And when you measure it, the cutoff for being therapeutic is you want to be able to deliver doses that will be low enough that it won't affect your bone marrow, but high enough to do those actions at least to 2 gray. And in Rex, we were getting up to 3 or 4 gray in all of his metastatic lesions and still sparing his bone marrow. And he didn't have any changes in his CBC over time after treatment. And toxicity, this particular drug, the NM600, is cleared through the biliary system. That's another advantage of it, actually. It isn't cleared by the urine, so this particular drug could be a theranostic for bladder cancer because we don't have to worry about a signal from the agent. So this is the trial that we have going. We're similar scenario, biospecimen rich. They go ahead and they get their yttrium 86, they get their dosimetry, then they get their yttrium 90. And then a few days later, we start injecting the intratumoral immunocytokine. Of course, again, timing, fractionation protocols, this is going to be a big learning process over the next 5 years to hopefully tease out and determine which fraction size, what the timing of the immunocytokine to the radiation should be, etc. But this is a true theranostic. Yttrium 86 can be used to stage these animals completely, and of course for dosimetry, and then yttrium 90 to have the therapeutic effect as well. Oh, and of course these patients are getting coarse fraction RT as well to their primary site, high dose to their primary site, as Rex did as well. So there's plenty of opportunities for basic and translational and clinical research in this area of radioimmunology. Just recently, just within the last 9 years really, if you look at the publication record of combining radiation with immunotherapy, this is all brand new. Lots of opportunity to come in. I'm just going to finish up. I've got about 6 minutes left here. We've been talking in this area here, immunotherapy. What about preventing cancer? Is that something that we could do? I mean, we talk about preventative vaccines like polio and distemper in the canine world. We can give it as puppies and never see that particular disease. Could we do the same with cancer? And that's something that certainly has been looked at for many, many years. And there are some prophylactic anti-cancer vaccines that are being investigated in people. We just started a trial based on technology that came out of Arizona State. Stephen Johnson heads up the Institute for Medical Advancement at Arizona State, and he's a big vaccinology guy. He's been around for several years doing vaccine work, primarily for infectious disease, of course. But over the last 5 years or so, we in Colorado State and UC Davis have been sending them serum samples and tumor samples from our cancer patients. And he's really been thinking outside of the box. This is a complete paradigm shift of, he feels that we've been looking under the wrong lamppost, looking at mutations in DNA. rather than looking at mutations that occur when RNA is translated into peptides and proteins, coming up with these frame shift mutations. And looking at all the samples that we sent, and he's also looked at the same thing in people and in rodents, finds a series of, in our vaccine, it's about 36 frame shift mutations, missplicing mutations, peptides that occur from that translational event that are conserved in cancer cells but not conserved in normal cells. So again, it's something that's relatively unique to the cancer cell, but the other thing is it's agnostic of tumor histology. So you can look for these frame shift mutations in women with breast cancer, men with prostate cancer, dogs with osteosarcoma, dogs with hemangiosarcoma, non-Hodgkin's lymphoma, histiocytic sarcoma, transitional cell carcinoma. We have looked at virtually everything that you can imagine. And based on this, we applied for the Department of Defense has something called high risk, high reward, which is, it's unlikely to work, but if it works, spectacular game changer. I mean, this is preventative medicine that is an inexpensive vaccine. It's not personalized, inexpensive, that could be applied globally in developing countries and third world countries, et cetera. So, and for example, there's a lot of critics. I mean, this is more likely not to work than work, right? It's outside, it's way outside the box. There was an editorial in Scientific American last summer, and the CEO of Cancer Centers of America said, there's no way this is going to work, but somebody has to do this trial, because even if there's a 1% chance of this working, this could be a game changer. So this is actually the largest interventional trial in the history of veterinary medicine. There's us, CSU, and UC Davis. We're entering 800 cases over 5 years. They come in, they get ultrasounds, chest x-rays, blood work. If they're deemed quote-unquote cancer-free, they have to be between the ages of 6 and 10. Then they're randomized to receive either the vaccine with the 36 frame shift proteins, peptides actually, or placebo, and then they're followed for 5 years. And they're vaccinated 4 times in a period of 8 weeks, and then every 6 months thereafter, and then yearly thereafter. And the endpoint is incidence of any cancer. And so even if you're in the placebo group, it's fully funded. You get 5 years of the best medicine, well, we feel the best medicine that a patient can. And if they develop a lump or bump, there's $2,000 towards diagnosis and potential therapy. So even if you're in the placebo group, we started enrolling in this. In 6 weeks, we had our 350 cases all in the queue, ready to go. So, and 4 weeks ago, some of you might remember Kara. She's our current clinical research intern. She's going to be one of our new residents coming in this year. This is the index case nationally. This made our state newspaper, made CNN, not this guy. I tried to keep this guy out of the picture because it's pretty rough. I really don't have time to go into the mechanics of it, but there are publications that you can look at. And the patients come in and they're vaccinated. We're using both a DNA vaccine and a peptide vaccine. So they get 2 DNA vaccines first, followed by 2 peptide-based vaccines. And then again, they're followed for 5 years. Couldn't do it without our patients. This was actually created by the lovely people at the Comparative Oncology Trials Consortium, which is the NCI group that coordinates all these veterinary clinical trials for cancer. And these are all of our patients. And somewhere in here, I know Fletcher's here, but I can never find it when I'm actually at that. But this was the rapamycin trial. These are all the dogs in the rapamycin trial. And this is not my group. I'm but a mere cog in part of this, but this is the veterinary group, our veterinary group at University of Wisconsin. But there's plenty of other players at the medical school involved in the trials that I mentioned. So, whirlwind tour of immunotherapy and the potential and the opportunity to include our companion species. And at the end of the day, we're veterinarians, so we want to get this information back into our clinics. If this vaccine, for example, technology works, it should readily be applied on the human side because the peptides are there, the immune system is very similar, so that would be the hope, but we could quickly certainly go into the veterinary side. We also are looking at using the same technology post-development of cancer. We have a hemangiosarcoma trial coming up using a subset of those particular frame shift proteins. So, you know, a lot of information in an hour. I just wanted to present some of the things that we've done that will illustrate how we can include our companion species in these trials and hopefully accelerate the process. So I'll be happy to entertain any questions or comments. Thank you. Yeah, Jim.
It's great talk. It's really exciting work. One of the things that interests me is the latency of metastatic disease. So you have the primary tumor, you have the initial onslaught of metastatic disease, but then following initial treatments, you can have dormancy for a long period of time. You can have senescent cells or quiescent cells. So how do you keep the pressure on, for that unforeseeable future?
Yeah, I mean, the hope is that if you're using an immunotherapeutic that obviously deals with the primary, that you create memory and that you can also continue that memory going in an effective fashion throughout so that you don't have immunosenescence and depletion of those particular T-cell subtypes, et cetera. I mean, that's a $1 million question. You know, melanoma is a classic example. You can be cured of melanoma and 20 years later it crops up. We have the potential in veterinary oncology to be really helpful because we have tumors that are, that show up without overt metastatic disease. We can deal with the primary, and down the road we have massive metastatic disease. So this whole idea of how do we apply immunotherapy in the minimal residual disease state and keep it recognized? I mean, the idea behind that vaccine against canine cancer study is to create the wanted posters, get them out there. We're using some of the new adjuvants that are in human clinical trials and maintain that and sustain that so that we create enough memory and enough robust memory. Now, obviously, none of these are going to work in a vacuum. Perhaps somebody will develop tumor down the road after a vaccination, and just the simple addition of a checkpoint inhibitor, all of a sudden their memory response will kick back in. I mean, we certainly saw that in some of the rodent models, that we can rescue a lot of some of the immunotherapeutics that we put on the shelf because they're just not helping enough people. We can start dragging those back off the shelf once we have, checkpoint inhibitors and some of these other technologies. So, I mean, it's extremely complex areas, and we're just scratching the surface right now, but it's a pretty exciting time. But then I was saying that 30 years ago, right? I was in vet school when I was doing space. It's great. It's awesome. Yeah?
Is there any consideration given to, in the VAX trial, bringing it into specific breeds like cancer?
Yeah, so I didn't show it because I don't have time, but we actually do limit the breeding. Mixed breeds are fine, and there's about 40 other breeds that are allowable. We cut off the low incidence breeds because we want more robust data. So we want dogs that are more likely to develop tumor. So there are inclusion criteria that are based on breed. So we've got, as you can imagine, we have clients out there whose pit bulls are becoming this or that or whatever. Pit bulls are eligible, by the way. We don't discriminate. We don't see breed in my clinic. Yeah. But just along those lines, if you look for those frame shift and missplicing peptides across breeds, it's agnostic of that as well. But, yes?
What sort of follow-up are you doing on the study?
Well, we're trying to make it a real-world situation. So for the first year, they come in at 6 months, primarily to collect PBMCs and serum and plasma. So the other advantage or the other potential from this trial is even if it fails, we are going to gain so much information on the canine immune system over a period of 5 years. Some of these things like the microvessel work, the extracellular, the EVs, collecting those over time and then looking, because there's really good markers of immunosenescence in those extracellular vesicles. But coming back around to your question, which I've had a transient ischemic event.
My question is what you're looking at for immune responses in these dogs.
Yeah, so we're looking at a lot of immune parameters, but are we going out of our way? Are we imaging every dog every 6 months for 5 years? No, that would be, I mean, $7 million is a lot of money. We would have to triple that. So it's real world. If a lump or bump is discovered, they have to be seen twice a year. full physical exam, how are things going. We use REDCap. I know you guys are starting to use REDCap, which is awesome. It sends emails out to the referring vets and to the clients, and it's time for you to go in for a physical and things. So we're being fairly strict about that, but yeah, it's as soon as something's found. And time to event really is irrelevant. It is incidence of cancer is the endpoint. Yes?
I work in a human cancer center.
Mm-hmm.
Mm-hmm. Talking about these type of trials and translating it into human trials.
Yeah, I mean, it's increasing over time. It's very rare. 20 years ago when I started going to ASCO, and I would have a poster or give a presentation, there's always somebody at the back of the room, a physician puts up their hand and go, well, so tell me again, how did you cause cancer in these dogs? I go, no, it's spontaneous. It's, this is, but after you've given your entire talk about it being a spontaneous tumor and whatever. And especially in 2015 when we had that, I'm trying to remember the name of what it's called, but twice a year at the National Academies, they pick a cancer topic, and they decided that it's so important that they should highlight the inclusion, I hate to use the word use, the inclusion of companion species in clinical trials. Could it work? What are the opportunities? What are the obstacles? How do we get around them? And it was a phenomenal group of physicians and veterinarians, the press, lay people, animal rights activists. Oh, I should have told you that we had one woman, you're only allowed to enter 2 dogs in a household to the vaccine trial. And we had a client come in last week with her 2 dogs. And she, we spent a lot of time on the phone explaining what's involved in the trial. But even so, she came in, and when she found out that we were giving 4 vaccines in 8 weeks, and then 1 at 6 months, and then every year thereafter, she goes, I'm an anti-vaxxer, that's too much heavy metals, and she wouldn't go into trial. So we've subsequently renamed this to the canine autism trial, but we're trying not to. But yeah, it's becoming much more of a, I talked about this CORC, this Comparative Oncology Research Consortium, so that's us in the Carbone, which is our center, Duke in North Carolina State, UC Boulder and CSU, UC Davis and UC Davis's Cancer Center, Minnesota and the Masonic, which is the University of Minnesota's Human Cancer Center, Pennsylvania and the Abrams, which is their cancer center. So these are all groups of, and again, we're literally forcing people to work together by saying you can't apply for this money. Unless you have a PI, a co-PI from the veterinary school and a co-PI from the cancer center. And the V Foundation, this is a huge group. Every year they have a fundraiser on ESPN. If you watch ESPN for a whole week, that's all they talk about. We were featured, and we're going to be featured in next year's. They've already raised, we had our first RFA for bladder cancer. And these are very large grants. They're very interested in the idea, the comparative approach. They've really bought in that generally the easy part is getting the individuals to realize it, the cancer patients and the families of the cancer patients to realize what the potential is. But yeah, we've come a long ways from. My pleasure. I like coming to the lesser veterinary school. Oh, I'm sorry. I didn't mean that. It's not true. Yeah.
I'm wondering if there is a stance that we know of regarding combination chemotherapy and immunotherapy, and whether chemotherapy and immune suppression can hinder the success.
Yeah, an excellent question. It's something that, for years we always said our chemotherapeutics are immunosuppressive. They're not. They're myelosuppressive. They can be immunosuppressive depending on how you give them. They're myelosuppressive, certainly. But in many cases, they're actually immunostimulatory. Doxorubicin, for example, at pulse dose, doxorubicin is an immunostimulant. We've shown that in dogs with bronchoalveolar lavages in dogs with osteosarcoma that we were giving toll receptor agonists and doxorubicin that we didn't reverse. We actually got more stimulation of our alveolar macrophages with chemo. So, but there's so many questions to be answered about timing and dosing that You know, we're early in that process. We're getting some hits where, I mean, the checkpoint inhibitors where 20% of people with stage 4 lung, stage 4 melanoma are having durable responses that are spectacular. But there's that 80% that we need to turn their cold tumors hot. And so that's what everybody is scrambling to try and do. The other problem with one of the reasons why radiation therapy really hasn't been brought into the mix with a lot of funding and a lot of publications is it's not patentable. You can't patent photons, right? So if there's no drug for a drug company to say, I want to do this, but in combination with some of the other drugs, yeah. But that's where the National Cancer Institute and, you know, the Moonshot, they're really putting a lot of emphasis on the inclusion of companion species to try and accelerate this whole process. So. All right, cool. Thank you.